Welcome to lecture 12 of the IMAT Biology series. I'm glad you're here because today we are going to cover one of the most beautifully engineered systems in the entire human body. We are talking about the renal system. And I want you to understand something before we even begin. This is not just a lecture about a pair of bean-shaped organs sitting behind your abdominal cavity. This is a lecture about a system that is simultaneously a filter, a pump, a pH balancer, a blood pressure controller, and a hormone sensing machine, all running in parallel all of the time without you ever thinking about it. And the IMAT examiners know this. That is exactly why this topic shows up on virtually every sitting of the exam. Over the course of this lecture journey, we will start with the gross anatomy of kidney, mapping out the cortex, medulla, pelvis, and ureter, before zooming down into the microscopic architecture of the nephron and its highly specialized functional segment. From there, we will unpack glomerular filtration by calculating the physical forces that dictate GFR and filtration fraction, followed by a deep dive into the mechanism of tubular reabsorption and regional specialization. We will also explore tubular secretion. We will master the intricacies of the countercurrent multiplication alongside the vasa recta and dissect the precise hormonal regulation involving ADH, aldosterone, RAAS, and ANP. Finally, we will close by analyzing how the kidney handles urine concentration and dilution, renal acid-base regulation, and fluid and electrolyte balance. This is a comprehensive roadmap, but mastering these subtopics will give you a massive point advantage on exam day. Let us begin our journey with our first core idea. To understand the fundamental overview of the renal physiology, we must map out the entire macroscopic and microscopic architecture from scratch on our canvas. We locate the primary components of the renal system arranged sequentially within the human torso. Torso, also known as the trunk, is the main central part of the human body. Imagine we cut off the head and both the arms. The remaining portion will be called torso. And we position the paired kidneys retroperitoneally along the posterior abdominal wall. In anatomy, retroperitoneal means behind the peritoneum, and peritoneum is the membrane lining the abdominal cavity. Imagine there was a membrane over here, but we removed that membrane just to see the kidneys clearly. Now moving downward from each kidney, the left kidney and the right kidney. If we move downward from here, we trace the long muscular tubes of the ureters which course inferiorly to enter the urinary bladder, a hollow muscular reservoir located in the pelvic cavity. Exiting the bladder inferiorly, we position the urethra, the final channel responsible for conducting urine out of the body. Now we zoom into the internal landscape of the kidney to isolate its true microscopic functional worker, the nephron. Imagine this is one kidney, and if we take a part or section of the kidney and zoom in, we will see structures like nephrons inside it. And each kidney contains approximately 1 million of these functional units working in parallel. We divide nephrons into two major populations based on their spatial positioning and structural layout. First, we locate the cortical nephrons, which make up 85% of the total population. Their renal corpuscles are positioned high in the outer cortex. Cortex is the outer part of the kidney. You can just think that this portion is cortex. If we go medially, we will have medulla over here, and further going will let us see pelvis, then ureter, and finally it will connect to urinary bladder. To clear confusion, I have written right kidney because I was showing only one kidney and it was on right side. So if we go from right side to left side, then the first part will be known as cortex, then leftwards it will be known as medulla, then gradually we will see pelvis, ureter, urinary bladder, and after that we will also see urethra that we have seen in the previous image. As an introductory side note, the renal corpuscle is the initial filtering unit of the nephron in the kidney, consisting of two main structures. First, the glomerulus, which is a tuft of high-pressure capillaries. This whole structure is called glomerulus. And the second part of renal corpuscle is Bowman's capsule. This cup-shaped structure is Bowman's capsule, and we will know about them more in detail in the coming sections. Second, we position the remaining 15% as juxtamedullary nephron. We locate the renal corpuscles deep in the cortex, directly adjacent to the cortico-medullary junction. The junction between cortex and medulla is called cortico-medullary junction. The juxtamedullary nephron possesses exceptionally long loops of Henle. This part you can consider loop of Henle roughly, and that plunge deeply into the innermost regions of the renal medulla, wrapping around specialized capillary networks. If you see the meticulous detailed image, then you will find the loops of Henle's are wrapped around by capillaries, kind of like this. As I have already shown you, the loop of Henle is the U-shaped portion of the nephron that conducts urine within each kidney. This deep penetration creates a critical anatomical layout that directly dictates the kidney capacity to generate concentrated urine and preserve vital body water. So to recap, we can tell human bodies normally have two kidneys, and in each kidney we will have around 1 million nephrons, and a nephron consists mainly of two parts. One is renal corpuscle, another one is renal tubule. We can further divide corpuscle into glomerulus and Bowman's capsule. And for tubule, we first will find proximal convoluted tubule, then loop of Henle. Moving further on will lead us to distal convoluted tubule, and finally the tubule portion will end with collecting duct. You don't need to worry, we will look and study all of the tubules in as much detail as it is required. As we finish mapping this structural arrangement from the gross organs down to the subpopulation of nephrons, we must realize that these physical boundaries directly drive the massive fluid dynamics of the body. The physical architecture of 1 million nephrons acting simultaneously establishes a high-capacity filtration bank, which immediately translates into massive daily volumes and homeostatic values we observed on our slide data. The renal system is structurally defined as the combination of the kidneys, ureters, bladder, and urethra. Its true microscopic functional unit is the nephron, numbering roughly 1 million per kidney. The system operates through 4 core physiological roles, which are filtration at the glomerulus, reabsorption along the tubules, secretion into the lumen, and final excretion from the body. Together, these processes maintain systemic homeostasis by tightly regulating water balance, electrolyte concentrations, arterial blood pressure, and plasma pH within a strict range of 7.35 to 7.45. And now looking at the core fluid dynamics, the normal human glomerular filtration rate is established at 125 milliliter per minute. That means the normal human kidney filters 125 milliliters of fluid out of the blood every minute. Remember we said glomerulus, Bowman's capsule? Bowman's capsule brings the blood, and the blood then gets filtrated by the rate of 125 milliliter per minute. And that is what glomerular filtration rate is known as. So we can say when blood arrives through the glomerulus, the filtering happens. And this equates to an astonishing total daily filtration volume of 120. The renal system is structurally defined as the combination of the kidneys, ureters, bladder, and urethra. Its true microscopic functional unit is the nephron, numbering roughly 1 million per kidney. The system operates through 4 core physiological roles, which are filtration at the glomerulus, reabsorption along the tubules, secretion into the lumen, and final excretion from the body. Together, these processes maintain systemic homeostasis by tightly regulating water balance, electrolyte concentration, arterial blood pressure, and plasma pH within a strict range of 7.35 to 7.45. And now looking at the core fluid dynamics, the normal human glomerular filtration rate is established at 125 milliliter per minute. That means the normal human kidney filters 125 milliliters of fluid out of the blood every minute. Just to remind you again, imagine these are the glomerulus. They come in contact with Bowman's capsule. Glomerulus brings in blood and the blood gets filtered. I'm showing only one nephron, but this process happens inside millions of nephrons we have in the kidney. To be more precise, around 2 millions in both kidneys together. And this filtration equates to an astonishing total daily filtration volume of 180 liters of fluid passing into the nephrons every single day. You might think the whole filtrate goes out as urine, but it's not what you think. 99% of the filtrate is reabsorbed. That means the filtrate of 180 liters do not get excreted as urine. This is not correct. Rather, 99% of the filtrate will get reabsorbed. How does it get reabsorbed? We will cover that in the tubular reabsorption section. When we said 99% of the filtrate is reabsorbed, it means that only 1.5 liters of fluid are actually excreted as urine each day. When this filtration and excretion system fails catastrophically in clinical practice, patients develop end-stage kidney disease, requiring a life-saving clinical procedure known as hemodialysis or definitive renal transplant. IMAT doesn't cover hemodialysis or renal transplant in details. That's why we will just know the names of the procedures, and that would be sufficient. Pay critical attention to these straps to protect your score. Examiners routinely deploy semantic swaps to confuse students on the true functional worker of the excretory system. A favorite trick question will state that the kidney is the basic functional unit of the renal system. You must remember that kidney is the gross organ, while the nephron is the true microscopic functional unit. Another major conceptual fallacy is assuming that the majority of filtered fluid is routinely excreted. The slide proves that the exact opposite is true. 99% of the massive 180 liters of daily filtrate is actively and passively reabsorbed back into the bloodstream, leaving only about 1% to be excreted as final urine. To secure top scores, don't forget about the exam pearls. You must commit these distinct anatomical percentages and their physiological adaptations to memory. Always remember that human kidney contains 2 distinct populations of nephrons. Cortical nephrons are the majority at 85%, whereas juxtamedullary nephrons make up only 15% of the total population. The second pearl states that juxtamedullary nephrons possess uniquely long loops of Henle that descend into the deep, highly concentrated medullary interstitium. This specific structural length is what gives juxtamedullary nephrons the unique power to drive the countercurrent mechanism, making them primarily responsible for urine concentration during states of severe dehydration. The topic of countercurrent mechanism will be covered in the following sections. Earlier, we got a superficial knowledge about the position of kidney and the microscopic structure of it. But if we want to master it, we must construct the entire organ from first principles on our conceptual canvas. We locate the paired kidneys in a retroperitoneal position on either side of the vertebral column. This is the vertebral column. We write VC for it. And the kidneys extend precisely from 12th thoracic vertebra to 3rd lumbar vertebra. Because of the massive space occupied by liver on the right side of the abdominal cavity, we position the right kidney slightly lower than the left kidney. And wrapping the surface of the kidneys, we place a tough external fibrous capsule. This whole part is the outer fibrous capsule, and this capsule maintains the organ shape and provides a protective barrier against physical trauma. Moving along the medial concave border, this side is lateral and this side is medial. So when we say we move medially, we're going from the left to right side, and moving medially, we locate a deep indentation known as the renal hilum. We have isolated renal hilum for better visibility. To be accurate in depiction, I'm drawing an outline to show the renal hilum. This region has indentation, and this critical structure works as a gateway for the primary entry for the renal artery, renal veins, and nerves. Actually, to correct your knowledge, we can say this renal hilum acts as the primary entry for the artery, but serves as the exit point for the renal vein, because we know artery enters an organ, but veins leave an organ. Also, lymph vessels will exit from the hilum. We haven't showed it in our diagram. Moving outward to inward across the parenchymal landscape, we divide the internal architecture into three major macrozones, which are the renal cortex, the renal medulla, and renal pelvis. We locate the granular outer cortex directly beneath the capsule, where the packed renal corpuscles, including the glomeruli and Bowman capsules, along with the proximal convoluted tubules and distal convoluted tubules, give this zone its characteristic granular appearance under a microscope. So to distinguish it better, we will put black ink, and the area shaded by black ink is the renal cortex. And if you look closely, you will see the grains visible, and we have intentionally made it so obvious for identification, as you guys don't need to know the nitty-gritty of renal structure. Now we're going to change the ink color to trace medulla, or let's say put the laser for distinguishing the medulla from cortex. Directly facing the deeper interior, we will establish the renal medulla, which is organized into 6 to 18 striated fan-shaped structures called renal pyramids. This one is a pyramid 2, 3, 4, 5, 6, 7, 8. It can fall in the range of 8 to 18. And remember, I mentioned the word striated. These are striations, the lines. And within this medullary zone, we position the thick limbs of the loop of Henle and the straight collecting ducts in the outer medulla. That roughly translates to this area. While the thin limbs of the loop of Henle, the papillary ducts, and the metabolic machinery for urea recycling are nested deep within the inner medulla. That translates roughly to this area. These are approximations. These are not accurate depictions, but I'm intentionally doing it for better understanding without any confusion. The broad bases, these are the bases, and these bases face the cortex, while their apical tips, these are the tips known as renal papillae, point toward the center to drain the newly formed urine. Surrounding individual papillae we locate a cup-like collection structure called minor calyx. For minor calyx, let's use MC. One minor calyx here, another one here, and the third one is over here. So we can tell multiple minor calyces merge into larger channels termed major calyces. So this will be roughly one major calyx, and they eventually empty into the large funnel-shaped renal pelvis. The renal pelvis then tapers as it exits the hilum. You see it's tapering here, and after tapering, the pelvis transitions directly into the muscular ureter to transport urine down to the bladder. The structural hierarchy dictates that the initial high-yield processes occur in the granular cortex, while the concentration gradient setups are restricted to the medullary pyramids, ensuring that urine collection concludes flawlessly at the central pelvis. As we complete the comprehensive spatial mapping from the external fibrous capsule down to the internal drainage pathways of the calyces, we see how the physical layout organizes separate functional zones. This macroscopic and microscopic orientation directly forces the fluid to travel along an unyielding pathway. And that brings us directly to a structured review of the core data points and potential testing vulnerabilities found on our current slide. The gross anatomy of the kidney is characterized by its retroperitoneal location along the posterior abdominal wall, with the right kidney situated lower than the left due to the anatomical presence of the liver. The medial border features the renal hilum, which serves as the main vascular and structural hub where the renal artery enters and the renal vein and ureter exit the organ. Internally, the kidney is organized into three distinct structural zones, which transition sequentially from the outer cortex to the deeper medulla and finally to the central collecting renal pelvis. The cortex exhibits a granular texture because it contains the glomeruli, proximal convoluted tubules, distal convoluted tubule, and Bowman's capsules. The medulla is structurally divided into outer and inner regions, consisting of 8 to 18 renal pyramids ending in the renal papillae. The outer medulla houses the thick limbs of the loop of Henle and collecting ducts, whereas the inner medulla contains the thin limbs of the loop of Henle papillary ducts and participates extensively in urea recycling. The path of urine drainage flows precisely from papillae into the minor calyx, emptying directly into the renal pelvis before traveling down the ureter. When severe large calculi or renal stones block this renal pelvis, or when malignant tumors destroy the parenchyma, some advanced surgical procedures such as nephrolithotomy or nephrectomy become necessary. When I was talking about the cortex and medulla, I covered the table, that's why I'm not repeating it. And that brings us to exam traps. One is written. I'm going to add some more, and you need to pay critical attention to these high-yield traps to protect your score from common examiner tricks. The first trap is attempting to misplace the components of nephron across the gross anatomical zones. Examiners will frequently write tricky true or false questions claiming that the loop of Henle is found within the renal cortex. You must remember this absolute structural rule. The loop of Henle is located in the medulla, not the cortex. The cortex contains only the glomeruli and Bowman capsules and the convoluted portions of the tubules, while the loop of Henle drops down into the pyramids to facilitate the concentration gradient. And to secure top scores, you must memorize the pearls that I'm about to share. You have to be able to memorize the direct correlation between the regional zoning and physiological function. The granular outer cortex is entirely specialized for the initial process of filtration, reabsorption, and secretion because it holds 100% of the filtration barriers. In sharp contrast, the outer and inner medulla are structurally optimized exclusively for concentration gradient. Or to be more precise, the outer and inner medulla are structurally optimized exclusively for concentration gradient setup and hyperosmotic environment maintenance. This medullary specialization is driven by the parallel arrangement of the long loops of Henle and papillary ducts, which work alongside urea recycling to maximize water conservation before the fluid ever reaches the minor calyces. Alright, now to understand the true functionality of nephron, we need to zoom into the nephron itself from the start to end in a sequence. And I want you to think of the nephron as a very long, twisting, purpose-built tube with different personality traits at different points along its length. It starts as a bulbous cup. This is the bulbous cup. Then it narrows into a twisting tube, dips into the medulla over here. From dipping, it comes back up and it twists again and then dumps into a shared collecting duct highway. This is the collecting duct, and each section along this journey has a specific set of channels, pumps, and transporters in its wall. And each section is selectively permeable to different substances. When I say selectively permeable, that means that the barriers will allow only certain substances to pass from one side to another. Now, IMAT loves to ask which substance is handled where, so you need to know each segment code. So first of all, we locate the beginning of this microscopic channel, the filtration headquarters, situated entirely within the renal cortex. Here we position the renal corpuscle, which is composed of the glomerulus. This tuft of capillary, as we have mentioned already, is called glomerulus. And you need to remember this glomerulus is composed of high pressure capillary bundles, and this glomerulus is wrapped tightly by the double-walled wall number 1, second wall, that's why double-walled Bowman capsule. This anatomical arrangement serves as the primary site of passive plasma filtration, allowing water and small solutes to move completely free into the tubule system while blocking large proteins and blood cells. That means only the smaller portions will come out from the glomerulus into the Bowman capsule, but the large molecules will get blocked. Now, exiting the renal corpuscle, the fluid moves immediately into the proximal convoluted tubule. This roughly represents proximal convoluted tubule. And this PCT, or proximal convoluted tubule, is also localized within the renal cortex. We map this segment with a highly active specialized cellular border featuring a 65 to 70% reabsorption rate, which acts as the massive reabsorption zone where the bulk of filtered sodium, chloride, bicarbonate, water, glucose, and amino acid are reclaimed from the filtrate. That means they will get into the bloodstreams. But hydrogen ions and organic acids are actively secreted into the lumen. That means from basolateral side, hydrogen ions and organic acids will get inside proximal convoluted tubule. And embedded within the apical membranes of this proximal segment, we position the sodium glucose co-transporter 2, known as SGLT2, which is completely responsible for reabsorbing glucose up to a strict glucose threshold of 180 milligrams per deciliter. If plasma glucose concentration exceeds this specific saturation limit, the transporters are overwhelmed, which directly will cause glucoseuria. Moving downstream, the architecture alters dramatically as the tubule turns vertically into the loop of Henle. So this marks the beginning of loop of Henle. This is the descending portion and this is the ascending portion. And the loop of Henle plunges straight down from the cortex into the renal medulla. This concentration gradient system is characterized by a stark asymmetry in descending versus ascending permeability. We first track the descending loop moving downward into the hyperosmotic medullary interstitium. When we say hyperosmotic, that means the osmolarity or the concentration of solute are higher over here. You don't need to be afraid of that word. And we map this specific segment, the descending part, with higher water permeability driven by aquaporin 1 channels, but it remains completely impermeable to solute, which allows water to exit via osmosis. That means water will come out from the lumen and it will go to bloodstream. And this progressively concentrates the luminal fluid. Luminal fluid means the fluid inside the tubule. So the concentration of the fluid inside the tubule will rise in this specific region. And at the sharp U-turn, this is the U-turn. In this sharp U-turn, the loop transitions into the thin ascending loop in the inner medulla, which allows passive sodium chloride reabsorption. Moving upward, the tube expands into the thick ascending loop. We will write TAL for this part, which is located in the outer medulla. We map this thick ascending segment as a strict water impermeability trap because its epithelial cells completely lack water channels and are non-negotiably impermeable to water. Instead, we locate the sodium-potassium chloride co-transporter or NKCC2 transporter. That means the transporters are embedded within thick ascending limbs like this, although it is magnified and shown just beside here. And this NKCC2 transporter actively pumps sodium, potassium, and 2 chloride ions out of the lumen into the medullary interstitium. This continuous solute extraction without water movement creates a highly dilute luminal fluid while establishing the steep medullary osmotic gradient. That means the osmolarity changes in this region of loop of Henle overall. Now we will emerge back into the renal cortex. The thick ascending limb of loop of Henle transitions into the final regulation zones, beginning with the distal convoluted tubule. Roughly, you can say the yellow shaded portions mark the beginning of distal convoluted tubule. In language, we locate this segment in the cortex and map it with the sodium chloride co-transporter or NCC. We are only showing one, but there are several. So many NCC transporters spread across the whole of distal convoluted tubule. And the sodium chloride co-transporter drives further sodium and chloride reabsorption. That means more sodium will get out from the lumen and chloride will also get out of the lumen and go into bloodstream. This NCC channel will be accompanied by specialized calcium channels regulated explicitly by parathyroid hormones for calcium uptake. Finally, the distal convoluted tubule form multiple nephrons empty into a single shared collecting duct as a site of hormonal fine-tuning, where the aldosterone acts to adjust final sodium reabsorption and potassium balance. An antidiuretic hormone also works over here, which binds to open some channels known as aquaporin 2 channels. Let's draw a few channels over here. Aquaporin 2. And this aquaporin 2 provide variable water permeability to control final urine concentration before excretion. As we finish tracking this microanatomical path from the cortical headwaters down to the medullary papillary ducts, we see how each segment's spatial layout directly dictates its transport capabilities. The mechanical trapping of water in the thick ascending limb is what establishes the concentration gradient necessary for the collecting duct to function under hormonal control, which brings us directly to our structured slide review to anchor these core parameters and transport events. The nephron is defined as the structural and functional unit of the kidney, composed of two main parts, which are the renal corpuscle and the renal tubule. The renal corpuscle is further subdivided into glomerulus and Bowman capsule. The absolute mechanism and sequence of fluid travel follows a strict linear pathway. Blood enters Bowman's capsule, moves through the proximal convoluted tubule, travels down and up the loop of Henle, passes into the distal convoluted tubule, and finally drains into the collecting duct, which aggregates fluid from multiple nephrons before terminating at the papillary duct. Let me remind you again, papillae. If I draw a renal pyramid, this is the base and this is the papillae, and there will be ducts which we know as papillary duct, and urine usually aggregates over here. You gotta be more precise. Don't just say urine, first say fluid. Now looking at the regional breakdown and transport events, the Bowman capsule and proximal convoluted tubule are located exclusively in the cortex, showing high water permeability, with the PCT utilizing aquaporin 1 channels to reabsorb 65 to 70% of filtered water and sodium, glucose, bicarbonate, amino acids. The descending loop of Henle is located in the medulla, maintaining high water permeability via aquaporin 1 to move water out and concentrate the fluid. The thick ascending limb of loop of Henle is located in the medulla, possesses absolutely zero water permeability, and utilizes the NKCC2 transporter to actively pump out solute, diluting the fluid. The distal convoluted tubule is located back in the cortex, possessing variable water permeability, and utilizes the MCC co-transporter along with parathyroid hormone regulated calcium channels, where aldosterone driven modifications begin. The collecting duct descends back into the medulla, possessing an ADH dependent water permeability regulated by aquaporin 2 channels, and uses epithelial sodium channels or ENAC for fine-tuning sodium reabsorption. When patients suffer from fluid overload or severe hypertension, clinicians routinely intervene pharmacologically using diuretic medications that target these transport proteins. Now coming to exam traps again. Pay critical attention here. The most heavily tested non-negotiable rule on medical admissions exams concerns the permeability of thick ascending limb. Examiners will constantly use distractors to make you believe that the thick ascending loop is permeable to water or that ADH controls it. You must remember this absolute truth. The thick ascending limb is completely impermeable to water under all physiological conditions. That's why written non-negotiable. Do not confuse this segment with the descending limb or the collecting duct. To secure top scores, you must memorize the key transport symbols and glucose thresholds. The proximal convoluted tubule possesses a strict maximum for glucose, known as the glucose threshold, which is established at 180 milligrams per deciliter. If a patient blood sugar spikes above this concentration, the transporters of the proximal convoluted tubules, the SGLT1 co-transporters, become completely saturated. That means they are not being able to pick up more glucose from the lumen, and that causes the excess glucose to remain trapped in the lumen, where it acts as an osmotic solute that prevents water reabsorption. That means glucose will be inside lumen and it will force water to stay with them, which explains the presence of glucose urea. That means glucose in urine and polyuria in diabetic patients. Now we will dive deeper into the glomerular filtration. And to master the mechanics of glomerular filtration, we must construct the entire microscopic filtration barrier and the mechanical pressure systems entirely from scratch on our conceptual canvas. First of all, we locate the structural gateway where blood flow enters the nephron through the renal arteries, which branch down into specialized glomerular arterioles. There are around 60 to 70 arterioles over here. Specifically, we position the white afferent arteriole conducting blood directly into the glomerulus, and this glomerulus is nested inside the cup-like expansion of the Bowman's capsule. The wider afferent arteriole enters, and the narrower afferent arteriole conducts blood away from this network. That means the arrow will point upward, not downwards that was originally given in the image. So we need to correct this concept. Now, if we take one capillary from here and we zoom into it, we will build three-layer filtration barrier from inside moving outward, and we will study it in this segment. First of all, layer 1 is the fenestrated capillary endothelium directly facing the passing blood flow. This endothelial layer features large pores. These are the pores that physically exclude red cells and platelets from entering the filtrate. Directly outside this endothelium, we position layer 2, the glomerular basement membrane, or GBM. This crucial middle matrix consists of a dense network that is intensely negatively charged, creating an electrostatic barrier that actively repels negatively charged serum proteins like albumin, thereby excluding them based on charge. Wrapping the outer surface of the GBM, we position layer 3, the porocyte filtration slits. Here, we locate specialized epithelial cell foot processes or feet that interlock precisely, creating narrow slits to exclude any remaining large proteins or macromolecules greater than approximately 70 kilodaltons from entering the tubule system. This triple-layered structural layout creates a strict molecular sieve dividing substances into what is and is not filtered. On the pass list of freely filtered substances, we place water, ions, glucose, creatine, uric acid, small hormones, and unbound drugs. On the excluded list of non-filtered substances, we place large serum proteins like albumin and globulins, RBCs, WBCs, and platelets. Also, we will exclude any large plasma protein bound substances. Now we superimpose the physics of ultrafiltration across this three-layer barrier by mapping the precise Starling forces acting within the corpuscle. We locate the final pressure favoring filtration by subtracting opposing pressures from favoring pressures. First, we establish the glomerular capillary hydrostatic pressure, written as PGC, which is a powerful outward force pushing fluid into Bowman space. That means this pressure tends to push fluid out of the blood vessel into the Bowman space, and this is measured at a high value of 60 millimeter of mercury. Opposing this forward movement, we position the Bowman capsule or Bowman space hydrostatic pressure, written as P subscript BS, which exerts a backward fluid pressure of 15 millimeter of mercury. Next, we locate the plasma oncotic pressure, written as pi GC, which represents the osmotic pull of trapped proteins retaining water inside the capillary, measured at 129 millimeters of mercury. Finally, we position the Bowman space oncotic pressure, written as pi bs, not rho, pi bs, which is normally 0 millimeters of mercury because proteins are excluded from the filtrate. So many of you get curious that why hydrostatic pressure pushes the fluid out and oncotic pressure pulls the fluid inward. The reason is pretty simple. Proteins pull fluid inward because of osmosis, where water naturally moves toward a higher concentration of solutes to create balance, while hydrostatic pressure pushes fluid outward because it is the physical mechanical force of blood pumping against vessel walls. Now, if we calculate the difference where 60 minus 15 minus 29 minus 0 occurs, we find a net filtration pressure, or NFP, of exactly 16 millimeter of mercury driving fluid forward. That means the net filtration pressure will drive the fluid out from the glomeruli into the Bowman space. And this pressure achieves a massive daily filtration volume, where the kidney filter the entire plasma volumes approximately 60 times a day at a glomerular filtration rate of 125 milliliters per minute, totaling 180 liters per day. And out of all the plasma entering the glomerulus, a precise 20% filtration fraction is successfully filtered into Bowman space. And to stabilize this matrix against daily blood pressure fluctuations, we locate autoregulation range between 80 to 180 millimeters of mercury, where myogenic autoregulation and tubulo-glomerular feedback sensed by macula densa cells measuring sodium chloride delivery dynamically adjust the afferent and efferent arterial diameters. That means these diameters can be modified based on pressure differences. And the word that I have said, macula densa, these are some special pressure sensing cells located in kidney. But if you want to be more precise, the macula densa actually works as a chemoreceptors that senses sodium chloride flowing through the kidney tubule. As we finish defining this intricate balance of mechanical layers and precise physical forces, we realize that any physical alteration to these pressures or the structural integrity of the barrier directly alters the chemical composition of the downstream urine. When the negative charges or physical slits of this sieve fail, the structural damage instantly changes the filtration parameters, bringing us directly to our structured slide review to anchor these core physiological matrix and diagnostic pearls. Glomerular filtration is defined as a passive ultrafiltration process driven across a specialized barrier by net filtration pressure. The mathematical formula for net filtration pressure is the quantity of glomerular capillary hydrostatic pressure minus Bowman capsule hydrostatic pressure minus the quantity of glomerular capillary oncotic pressure minus Bowman space oncotic pressure, yielding a normal baseline value where 60 minus 15 minus 29 minus 0 equals 16 millimeters of mercury. The system maintains key baseline parameters, including a normal glomerular filtration rate of 125 milliliters per minute, which equals an absolute daily filtration volume of 180 liters of fluid per day. The filtration fraction represents the exact percentage of renal plasma flow that passes into Bowman space, established normally at 20%. That means filtration rate divided by total blood flow. For simplicity, the filtration barrier consists of three distinct structural components, which are the fenestrated endothelium, the glomerular basement membrane, and the porocyte filtration slits, with the GBM being heavily negatively charged to actively repel serum proteins like albumin. Freely filtered substances that enter the tubule system include water, iron, glucose, amino acids, urea, creatinine, uric acid, small hormones, and drugs, while excluded substances include albumin, globulins, red blood cells, white blood cells, platelets, and protein-bound substances. Now pay critical attention to these traps. Examiners love to test your conceptual understanding of how systemic alterations change the net filtration pressure through mathematical manipulation. A classic exam trap describes a patient with severe liver cirrhosis or malnutrition who has lost the ability to synthesize plasma proteins, leading to low plasma protein level. The examiner will ask you how this affects filtration. You must recognize that low plasma proteins mean a drop in the plasma oncotic pressure, which significantly reduces the opposing force. Consequently, the overall net filtration pressure increases, causing a paradoxical increase in the initial filtration rate. Another vital trap is identifying protein in the urine, known as proteinuria. The presence of protein in the urine is never a normal secretion event, but rather serves as a major red flag indicating physical or electrostatic damage to the triple-layered filtration barrier itself. To secure top scores, you must memorize the ideal properties of key clinical markers. Serum creatinine serves as the absolute ideal proxy or marker for tracking kidney function and GFR in clinical medicine. This holds true because creatinine is an endogenous molecule that is freely filtered across the three-layered barrier, and it is completely unabsorbed by the subsequent tubule segment and is only minimally secreted. Therefore, there is a strict inverse relationship between GFR and plasma creatinine levels. As GFR falls due to worsening renal failure, plasma creatinine levels must automatically rise, providing urologists and nephrologists with an immediate, reliable warning of declining renal clearance. Of the 180 liters filtered per day, the body only excretes about 1 to 1.5 liters as urine. That means the kidneys have to reabsorb approximately 99% of everything that was filtered. And that brings us to the process of tubular reabsorption. To master the precise cellular dynamics of tubular reabsorption, we must build the entire transcellular transport machinery and biochemical path completely from scratch on our conceptual canvas. We define this physical process as the directional movement of fluid and solutes from the tubular lumen. That means if it's tubule, this is the lumen of the tubule, and around tubule you will have capillaries that will wrap around them, and they are called peritubular capillaries. So tubular reabsorption is the physical process where directional movement of fluid and solute from the tubular lumen back into the peritubular capillaries occur. That means the fluid and solute will go from lumen back to capillary. Let's say this is the tubule and this is capillary. We always try to draw the schematic diagram for simplifying the concept. We divide water reabsorption into two distinct physiological classes, which are obligatory water reabsorption, making up 85% of total recovery, taking place constitutively within the proximal convoluted tubule, and descending loop of Henle. On the flip side, we have facultative water reabsorption, making up remaining 15%. So obligatory reabsorption constitutes 85% and facultative reabsorption constitutes 15%. And to drive this massive reclamation, we must place the absolute metabolic engine of the entire nephron on our canvas. We locate the primary engine, the sodium-potassium ATPase pump, embedded densely along the basolateral membrane of the epithelial cells. That means if this is the apical membrane, then this membrane will be basolateral membrane. Or we can say the portion of a cell's plasma membrane that faces adjacent cells and the underlying extracellular tissue are called basolateral membrane. Let me remind you from the digestion lecture, if this is a tube or tubule and if we have cells over here facing the lumen, then this membrane is apical membrane and the membrane opposite to the apical membrane is considered basolateral membrane. This is basolateral and this is apical membrane. So the sodium-potassium pump or sodium-potassium ATPase is situated on the basolateral side or basolateral membrane, not on the apical membrane. Watch out for that mistake. So understanding that concept or reviewing it, let's put one model sodium-potassium pump over here. Let's say this is our sodium-potassium pump. The sodium-potassium ATPase continuously expands adenosine triphosphate to pump sodium out into the interstitium while bringing potassium inside. If this is a cell and if we have sodium-potassium pump here, it will pump out the sodium and it will pull in potassium. 3 sodiums will go out and 2 potassiums will get in. And thus sodium-potassium pump maintains low intracellular sodium concentrations and highly negative electrical potential inside the cell. When you see that voltage of a particular cell is minus 60 millivolt or minus 70 millivolt, it means that the inside of a cell is negative or hyperpolarized or just polarized. And when we have negative inside and positive outside, it creates a steep gradient, and that gradient serves as the vital driving force for almost all secondary active transport occurring across the apical border facing the lumen. Now we move sequentially down the subsegments of the nephron to map every major apical channel. First, we locate the workhorse, the proximal convoluted tubule or PCT, this part particularly, which achieves a massive 65 to 70% obligatory reabsorption of the filtered load. On its apical membrane, we position a highly specialized family of co-transporters utilizing that sodium gradient. We place the sodium glucose co-transporter 2 or SGLT2. Let's draw one here. Let's say this is SGLT2, and this SGLT2 couples sodium influx to pull in glucose. That means this SGLT2 pulls sodium and glucose also follows that sodium behind it. Thus, proximal convoluted tubule pulls glucose from the tubule with the help of SGLT2. Beside it, we place the sodium phosphate co-transporter known as NAPI and also separate amino acid co-transporters, let's say over here, to completely clear these vital nutrients from the lumen. To manage acid-base balance, we also position the sodium hydrogen exchanger isoform 3 or NHE3. Let's put NHE3 over here, which pumps a hydrogen ion out into the lumen in exchange for a sodium ion moving in. To allow water to follow these reabsorbed solutes passively and constitutively without any hormonal control, we line both the apical and basolateral membranes of the PCT with aquaporin 1 channels known as AQP1. We have already seen it, that's why we're not drawing it again here, to keep the picture as tidy as possible. Now if we move down into the medulla, the fluid transits the descending loop. This is the descending loop. First, the fluid transit descending loop before climbing back up into the thick ascending limb. This is the thick ascending limb or TAL. And this TAL acts as our diluting segment. Here we position an epithelial border that is absolutely non-negotiable and strictly impermeable to water. That means water cannot get in, neither get out, because in this thick ascending limb, we don't have any water channel. Instead, we locate the sodium-potassium 2-chloride cotransporter, or NKCC2, which leverages the gradient to pull one sodium, one potassium, and 2 chloride ions out of the lumen simultaneously. This leaves the water trapped behind, creating a highly dilute luminal fluid while packing the medullary interstitium with solute. That means the fluid inside this portion has become quite diluted. Now, emerging back into the cortex, we transition into the distal convoluted tubule. This is our DCT, or distal convoluted tubule. And when fluid comes here, it undergoes regulated recovery. On the apical side of DCT, we locate sodium chloride cotransporter, or NCC. Remember, the cotransporter doesn't embed like an integral protein here. It will be only in one side, but this transporter has been exaggerated for understanding over here. So imagine that this is NCC cotransporter, and this NCC drives further neutral salt uptake. And for calcium homeostatic adjustments, we position TRP V5, or transient receptor potential vanillate member 5 channel, which is stimulated explicitly by parathyroid hormone and calcitriol to drive transcellular calcium reabsorption. Finally, the tubule system terminates at the collecting duct, which acts as the final adjuster. Here we locate principal cells utilizing epithelial sodium channels, or ENAC, to absorb sodium under the direct control of aldosterone hormone. To regulate the final 15% of the facultative water recovery, we position intracellular vesicles containing aquaporin 2 channels. When antidiuretic hormone, or ADH, binds its receptor, it triggers the precise trafficking and insertion of these aquaporin 2 channels directly into the apical membrane. Let's say ADH comes, ADH binds, and the channels will open. As simple as that. You don't need to think complexly about the mechanism. Just think that hormones will come, it will bind to receptor, and the channels will open, and that will allow water to leave the lumen and enter the peritubular capillaries. Remember, the tubules are not alone. Capillaries will wrap around the tubules like this. As we finish defining this intricate network of secondary active cotransporters and variable water channels, we see how the basolateral engine directly powers every single apical entry step downstream. The mechanical limits of these specialized proteins mean that if a solute concentration rises too high, the molecular channels hit an absolute physical wall, which brings us directly to our structural side review to anchor those core definitions, diagnostic traps, and therapeutic breakthroughs. Tubular reabsorption is defined as the physiological movement of water and solutes from the tubular lumen into the peritubular capillary network. The system is split into obligatory reabsorption, which represents 85% of total fluid recovery, occurring constitutively in the PCT and descending loop, and facultative reabsorption, which represents 15% of water recovery taking place in the collecting duct depending on hydration and ADH levels. The entire system is powered by sodium-potassium ATPase pump, which maintains low intracellular sodium to drive secondary active apical cotransporters. Looking at the precise regional breakdown, the PCT apical membrane utilizes SGLT2 for glucose, NAPI for phosphate, specialized amino acid cotransporters, and NHE3 for sodium hydrogen exchange, alongside constitutive aquaporin 1 water channel. The thick ascending limb utilizes NKCC2 cotransporter to pump out sodium, potassium, and chloride without any water movement. The DCT utilizes the NCC cotransporter for salt reabsorption and TRPV5 channels for parathyroid regulated calcium uptake. The word uptake means calcium is being reabsorbed into the blood. The collecting duct uses epithelial sodium channel or ENAC channels for sodium fine-tuning under aldosterone control and aquaporin 2 channels for hormone-regulated facultative water reabsorption, hormone being ADH or antidiuretic hormone. Fun fact, ADH is also known as vasopressin because ADH raises blood pressure. When patients present with severe volume retention, chronic kidney disease, or type 2 diabetes mellitus, doctors target these transport pathways using highly specific medications. Pivoting our path to traps, and the first trap involves examiner trying to test your understanding of what obligatory water reabsorption actually means. Examiners will try to convince you that water reabsorption in the proximal convoluted tubule is regulated or altered by antidiuretic hormone. You must remember this absolute truth. Obligatory water reabsorption via aquaporin 1 is entirely constitutive, meaning it happens automatically and is completely independent of ADH. Another major trap involves the limits of glucose reabsorption. Examiners will state that kidney can reabsorb an infinite amount of glucose. In reality, the SGLT2 transporters hit a strict transport maximum when the filtered load exceeds approximately 375 milligrams per minute, though plasma levels above the renal threshold of 180 milligrams per deciliter will cause individual nephrons to saturate, causing excess substrate to remain trapped in the lumen and spill into the urine as glucosuria. If you want to be in the top percentile, you must memorize the therapeutic mechanism and outcomes of SGLT2 inhibitors or gliflozins. These pharmacological agents function by selectively blocking the SGLT2 cotransporter in the proximal convoluted tubule, which intentionally lowers the transport maximum for glucose. By preventing this nutrient reclamation, the drugs induce a state of therapeutic glucosuria. This intentional spill of sugar into the urine not only lowers systemic blood glucose levels in diabetic patients, but also provides a mild osmotic diuretic effect that reduces blood pressure and protects the cardiovascular system, making it a critical standout drug class for medical admission boards. And this transport maximum mechanism, we already have said, there's a limit of glucose reabsorption that is done by proximal convoluted tubule. If it exceeds that limit, then the excess substrate will spill into urine because a transporter can only reabsorb so much glucose. If it exceeds the limit, it cannot do it anymore and it will spill glucose into urine, which will manifest as glucosuria. Filtration is passive and non-selective within its size and charge limits. Reabsorption reclaims what the body, but there's a third process that makes the kidney truly exceptional at clearing the blood, and that is renal tubular secretion. Secretion is the movement of substances from the peritubular capillary blood or from the tubular cell into the lumen. So it can come from peritubular capillary blood or cell to lumen, and this is the lumen. We already know the lumen is the hollow, empty, tube-like space inside the tubule. And what's the purpose of this renal tubular secretion? They come into the lumen so that they can be excreted in the urine. If I draw the picture, or let's say if I just show you the path, then the fluid will keep flowing like this, and it will travel this whole tubular path, and finally it will get out of the body as urine. It is an active selective process that allows the kidney to eliminate things that are too large to pass through the glomerular filter, or that are bound to plasma proteins and therefore excluded from the filtrate, or that need to be cleared from the blood more rapidly than filtration alone can achieve. We locate the primary clearance hub situated entirely within the renal cortex, and we will target the proximal convoluted tubule, roughly this region. And this proximal convoluted tubule is the site of organic ion secretion. I have drawn a schematic diagram to demonstrate how this process occurs. So I have just shown a sample of peritubular capillary or blood vessel beside the renal tubule. Let's say this is a segment of proximal convoluted tubule. And we all know tubule has tubular cells. There are so many cells, but I have only drawn one with all the details required for our purpose. One thing to clarify, you don't need to remember the specific ion channel names for the exam. If it is required, then I will tell you otherwise. So one tubular cell is shown here, and this part is apical site, and this part is basolateral site. A quick note, basolateral site will face the blood vessel or interstitium, and apical site will face tubular lumen. You can imagine there's a lumen going like this, and if you zoom down to the basolateral site of this PCT epithelial cells, we will position the organic anion transporters, specifically OAT1 and OCT2. You don't need to remember the names of them, just you need to know that there are organic anion and cation transporters on the basolateral membrane of the tubular cells, and their job is to actively take up organic ions. If we talk about the anions are handled by OAT1 and OAT3 transporter. They actively take up organic anions like penicillin and NSAID drugs from blood into the cell. So this penicillin will get inside the cell with the help of OAT1 and 3 transporters. And the organic cation transporters will do the same for cationic drugs and creatinine, as it is also positively charged. Now, if we directly go across the cell on the apical membrane, this is the apical membrane, we position MRP2 or MRP4 transporters, which will finally eject the anions from the cell out to the lumen. That means the anion will eventually get out of the body through urine. Now we map the specific ions and chemical entities handled across these distinct segments. I mean the segments of proximal convoluted tubules. There will be different transporters across the PCT. Now for hydrogen ion secretion, we locate active transport working simultaneously in both PCT and collecting duct. Let's say this is Bowman's capsule. Then we have PCT going like this. Then we will have loop of Henle, then TCT, and finally collecting duct. So first complete the PCT component for hydrogen ion secretion. In the PCT cell, this left portion, this portion represents the PCT cell mechanism. And inside the cell, I have taken only one cell, and we locate the sodium hydrogen exchanger isoform 3 or NHE3. Don't worry about the names. Just know that there's a transporter in the PCT cells, and they actively pump hydrogen from the cell to the lumen. And now moving on, we have come to the collecting duct. In the collecting duct, we will have alpha intercalated cell. And in the apical membrane, we position two distinct molecular engines. The first one is hydrogen ATPase pump, where the hydrogen ion is actively pumped into the lumen. You see this hydrogen is getting pumped out from the cell to the lumen. Remember, this side is the luminal site, and in exchange of that, the cell is receiving one sodium. And just beside that, we have hydrogen potassium ATPase pump, which exchanges one hydrogen for one potassium. You see the direction? Hydrogen is getting out and potassium is getting in. Now there's a problem. When we dispatch so much hydrogen ion to the luminal site, there's a high risk of the luminal pH becoming too toxic. And for that, we need to protect the acidity. That brings us to the ammonium buffer trap. Don't get confused. We're still in the collecting duct. What happens here is cells will metabolize glutamine to generate neutral ammonia gas, NH3. They will then diffuse freely across the lipid bilayer into the lumen. This is the luminal site, so we will write lumen for this. And this is the basolateral site. Let's just write B for it. Once the ammonia is inside the lumen, this ammonia instantly binds with the proton that was already present in the lumen, and it makes ammonium ion. This lipid bilayer is entirely impermeable to charged ions. That's why this ammonium ion becomes physically trapped within the lumen and is successfully excreted from the body, serving as the dominant acid excretion method that allows the kidney to handle large acid loads while respecting the absolute minimum urine pH threshold of approximately 1.4. Now we're moving to the potassium handling part. First, we locate the primary regulation site within the principal cells of the collecting duct. This is a principal cell, and this principal cell is located in the collecting duct portion of nephron. And this principal cell operates via a tightly coupled sodium-potassium seesaw mechanism. On the basolateral membrane, we have sodium-potassium ATPase. This transporter, or pump to be more precise, is called sodium-potassium ATPase. The sodium-potassium ATPase pulls in potassium and throws out sodium out from the cell. Thus, sodium-potassium ATPase builds a high concentration of potassium inside the cell. And on the apical membrane, we position two pathways working in tandem. This is the apical membrane. First one is ENAC, or epithelial sodium channel. The second one is renal outer medullary potassium channel, or ROMK. When the hormone aldosterone binds to the receptors, this aldosterone drives a precise sequence. It upregulates the insertion and activity of ENAC channels. And this ENAC brings sodium ions rapidly into the cell. And the sodium comes from the lumen. And this sodium reabsorption alters the electrical potential across the membrane. That means on the luminal side, there will be negative charge. And this negative luminal charge mechanically pulls the positive potassium ions that was generated through the open ROMK channels, resulting in active potassium secretion into the final urine. Finally, deep within the medullary collecting duct, we position the urea transporters UTA1 and UTA3, which actively secrete urea into the deep medullary interstitium to maintain steep hyperosmotic medullary gradient. Finally, deep within the medullary collecting duct, we position the urea transporters UTA1 and UTA3. UTA1 pulls the urea from the lumen into the cell, and UTA3 pulls the urea from the cell into the medullary interstitium. And this process maintains the steep hyperosmotic medullary gradient. Some people might think, wait, it was supposed to be secreted, but why it is getting reabsorbed again? And if any one of you get that thought, you are completely right that it's getting reabsorbed, but eventually it will get excreted in the urine. In fact, this urea is the primary nitrogenous waste product that the human body eliminates. However, the kidney manages urea through a fascinating process called urea recycling, which acts like a highly efficient recycling loop rather than a simple one-way garbage chute. That means the urea, first of all, will come to the medullary interstitium. It will do some job to maintain the osmotic pull, and after urea does its job, it finally gets excreted in the urine. That means urea will revolve in a route. Finally, deep within the medullary collecting duct, we position the urea transporters UTA1 and UTA3. UTA1 pulls the urea from the lumen into the cell, and UTA3 pulls the urea from the cell into the medullary interstitium. And this process maintains the steep hyperosmotic medullary gradient. Some people might think, wait, it was supposed to be secreted, but why it is getting reabsorbed again? And if any one of you get that thought, you are completely right that it's getting reabsorbed, but eventually it will get excreted in the urine. In fact, this urea is the primary nitrogenous waste product that the human body eliminates. However, the kidney manages urea through a fascinating process called urea recycling, which acts like a highly efficient recycling loop rather than a simple one-way garbage chute. That means the urea, first of all, will come to the medullary interstitium. It will do some job to maintain the osmotic pull, and after urea does its job, it finally gets excreted in the urine. As we finish defining all the concepts in our structured slide exams, renal secretion is defined as physiologic of substance from blood direct into which efficient, clear, large, protein-bound or rapid from the systemic circulation, operates via highly specialized sites, hydrogen ion in both the and the collecting duct using the NH or X. The hydrogen ATPase pump, and the potassium for systemic phase regulation. Potassium exclusively in the principal cells of the collecting duct through ROM, driven directly by the sodium channel sodium reabsorption. Ammonium synthesized from in the proximity via NH, where ammonia is for hydrogen ion to facilitate ionic trapping. Organic anion, including many common drugs, treated in the basolateral and transporters and apical MR pumps, while created cleared via organic cation transport in the. Looking at the mechanism, aldosterone acts on collecting duct, and the cells of the collecting duct upregulates epithelial sodium channel, and as a result, sodium gets inside, and ROMK upregulation causes potassium to get out and get excreted as urine. And as we were talking about hydrogen ion being toxic in too much amount, hydrogen ion gets added with ammonia, which becomes ammonium in the tubular lumen, and it cannot get inside the cells anymore because charged ions cannot cross the phospholipid bilayer, and thus the acid is excreted by this fashion, keeping our pH levels in check. Now going to the traps. First one talks about aldosterone. Examiners love to test the sodium-potassium seesaw by stimulating states of primary hyperaldosteronism, or also known as Cone syndrome. In this case, aldosterone increases, and the classic exam trap will try to trick you into thinking that excess aldosterone causes hyperkalemia. You must remember the non-negotiable mechanism. Aldosterone drives epithelial sodium channel up, as we already said, which forces more sodium reabsorption, naturally forcing more potassium out through ROMK channels. Therefore, hyperaldosteronism always leads to severe potassium loss, resulting in diagnostic hypokalemia. Another major trap involves misidentifying spironolactone. Examiners will try to state that all diuretics cause potassium wasting. You must boldly counter this fallacy. Spironolactone is a direct aldosterone antagonist, making it a classic potassium-sparing diuretic that protects systemic potassium levels. That means diuretics usually causes the loss of potassium ions from the body through urine, but this specific drug, spironolactone, it doesn't cause the potassium loss like the other diuretics. That's why this spironolactone is known as potassium-sparing diuretic. And to secure your top score, you must memorize the chemical boundaries of ammonium buffer trap and its absolute limit. The human kidney cannot physically lower the urine pH below an absolute minimum threshold of approximately 4.4 because of this steep electrochemical gradient. Secreting free hydrogen ions alone is completely insufficient to clear the massive daily acid loads produced by protein metabolism. The definitive example is that ammonium buffer trap is entirely essential for large acid loads. The nephron effectively masks the free hydrogen ions, allowing the kidney to excrete massive amounts of metabolic acid without violating the absolute minimum pH limit of 4.4. Now we arrive at the most elegant piece of engineering in the entire kidney, and honestly one of the most elegant in all of physiology. The question is this: how does the kidney produce urine that is 4 times more concentrated than blood plasma? The plasma osmolarity is about 285 to 295 milliosmoles per kilogram. The maximum urine osmolarity the human kidney can achieve is about 1200 milliosmoles per kilogram. That is a 4-fold concentration, and it is made possible by a mechanism called countercurrent mechanism, and it happens in the loop of Henle. To decode the intricate mechanism of urine concentration and dilution, we must map the entire spatial layout of the countercurrent architecture from first principles on our conceptual canvas. We locate this physiological machine functioning within the renal medulla, where 2 specialized vascular and tubular loops work in perfect symmetry to manipulate water and solute transport independently. These loops are, first of all, the loop of Henle, acting as the definitive countercurrent multiplier, and 2 being vasa recta, serving as the countercurrent exchanger. Together, they establish and preserve a steep hyperosmotic medullary interstitium that transitions smoothly from a normal isotonic concentration of 300 milliosmoles per liter at the cortical border down to an extreme hypertonic value of 1200 milliosmol per liter deep within the medullary pyramids. We will build the countercurrent multiplier concept by tracking how the fluid flows through the hairpin loop of Henle. This is the descending part of loop of Henle, then this bend, this is the hairpin loop of Henle, then this is the ascending loop of Henle. To get a clear picture, let's look at this diagram. The fluid enters the thin ascending limb of loop of Henle entirely isotonic. That means the concentration of the fluid inside the descending loop of Henle is same as the concentration of plasma. As it moves vertically downward into the increasingly salty medulla, it faces an epithelial wall that is highly permeable to water. This specific site, the descending thin loop of Henle, has some channels that draw out water from it, but it's completely impermeable to sodium chloride. And because of this structural reality, water is continuously drawn out. The water goes from the lumen into the hypertonic interstitium, and the process is osmosis. And this progressively concentrates the tubular fluid until it reaches its maximum concentration of 1200 milliosmol per liter at the very tip of the loop. Now the fluid rounds the sharp bend. This is the bend we are talking about. And it begins climbing upward through the ascending limb. This is the ascending limb. Don't get confused. These are the blood vessels, but this is our track of the loop of Henle. This was the descending part. This is the hairpin portion. Then we have ascending portion. And in this region, the structural design completely reverses. The epithelial wall of the thick ascending limb. This epithelial wall of the thick ascending limb is entirely non-negotiable and strictly impermeable to water. Instead, its apical membranes are packed with the sodium-potassium 2 chloride transporter, or NKCC2 transporter. This NKCC2 transporter pumps sodium, potassium, and chloride ions out of the lumen into the surrounding tissues. Because of this, massive quantities of solutes are actively extracted while water is mechanically trapped inside the lumen. The fluid becomes progressively diluted. We were talking in the deep medullary interstitium, the osmolarity is very high, 1200, but as it climbs up again, the osmolarity decreases. It becomes 400, 600, something like that. And from thick ascending limb, the fluid comes to the cortex part as a highly dilute hypotonic fluid measuring approximately 100 milliosmol per liter. Let's say the fluid is right now in this space and the osmolarity will be around 100. This active deposition of salts into the tissue without water is what continuously multiplies the concentration gradient, building the high medullary osmolarity from scratch. So from our understanding, we can level up some regions here. As the fluid goes down, the osmolarity increases until it reaches the peak of 1200 milliosmol per liter, and it needs to be preserved. And for that purpose, we introduce vasa recta. And vasa recta works as countercurrent exchanger. We position this slow-flowing hairpin-shaped capillary. You see, it has become hairpin-shaped. And these hairpin-shaped capillary loops run directly parallel to the loop of Henle. As the descending vasa recta, this is the descending portion, as this descending vasa recta blood flows down into the hypertonic medulla. Hypertonic means the osmolarity is more than the osmolarity of blood or plasma. When the fluid flows down into this area, it passively gains solute and loses water. That means it will pick solutes inside it, but it will lose the water it has. However, as the capillary loops around and becomes the ascending vasa recta, this part, the process completely reverses. It passively regains water from the interstitium and sheds its excess solutes back into the medulla. This passive, sluggish exchange allows the blood vessels to nourish the medullary tissue while leaving the overall hyperosmotic gradient perfectly intact. Finally, we will position the medullary collecting duct. The part of the collecting duct situated in the medulla is called medullary collecting duct, and this medullary collecting duct is the final valve determining whether the body excretes concentrated or dilute urine. And this decision is dictated entirely by a hormone called ADH, or anti-diuretic hormone. To understand what's happening here, let's zoom in and make a bigger picture. Let's say this portion is medullary collecting duct, and let's say these are the cells of medullary collecting duct. We will have some receptors for vasopressin on the vasolateral side of the cell. Let's say here's a receptor for vasopressin. When the body is severely dehydrated, high ADH levels spark an intracellular signaling cascade that forces vesicles to traffic and insert aquaporin 2 channels, or AQP2. Let's say one here. These are AQP2 channels, and they are in the apical membrane. You need to remember the inner medullary collecting duct is also highly permeable to urea via urea transporter A1 channels that we discussed. Because of this, urea actively exits the lumen alongside water. That means water gets inside and urea also does the same thing. That means it gets inside from the lumen into the cell. And this urea, when comes into the interstitium, contributes a massive 50% of the total medullary solute gradient. Under these conditions, water flows rapidly out of the collecting duct into 1200 milliosmol per liter interstitium, leaving behind a highly concentrated, low-volume urine. Conversely, during states of water excess or hyperhydration, ADH synthesis is completely shut down, and the receptor that was there for ADH, it doesn't get the ADH anymore. And without ADH, the aquaporin 2 channel we discussed, this aquaporin 2 channel synthesis is completely shut down, and as a result, the collecting duct becomes entirely impermeable to water. The dilute fluid entering from the cortex, let's say fluid is entering, coming to collecting duct, this dilute fluid keeps flowing downward without any water escaping, and it culminates in the excretion of a high volume, highly dilute urine measuring as low as 50 to 100 milliosmol per liter. As we complete this detailed mapping of the active multiplying transporters and passive vascular exchanges, we see how the structural layout of these paired loops creates a perfect osmotic trap. The mechanical exclusion of water in the thick ascending limb is what directly provides the chemical driving force for water reclamation in the collecting duct. This is the collecting duct. And all this discussion leads us directly to our structured slide preview to anchor these core concepts. By definition, countercurrent multiplication is responsible for creating deep corticomedullary osmotic gradient, which increases dramatically from 300 to 1200 milliosmoles per kilogram as we move deeper into the renal medulla. In order to successfully establish this gradient, the system strictly requires three structural features. First, a parallel arrangement of fluid flow, selective regional permeability, and active transport pumps. To understand the sequence of this mechanism, we must begin out of anatomical order with the thick ascending limb. Here, the NKCC2 transporter actively pumps sodium, potassium, and chloride ions out of the fluid into the surrounding tissue. But because this segment is strictly impermeable to water, no water follows the solute. Consequently, the surrounding medullary interstitium becomes hypertonic. This hypertonicity directly drives the next step in the descending limb, where water exits the permeable tubule by osmosis into the highly concentrated interstitium, causing the remaining tubular fluid to become increasingly concentrated. By the time this fluid reaches the very bend of the loop, it achieves its most concentrated state at approximately 1200 milliosmol per kilo. As this fluid ascends back toward the cortex, it gradually dilutes once again as sodium chloride exits, ultimately delivering a highly dilute fluid to the distal convoluted tubule. When preparing for your exams, there are two critical traps to avoid. First, remember that while the loop of Henle creates this essential osmotic gradient, it is the adjacent network of the vasa recta that preserves it. Second, be aware that the loop diuretic furosemide explicitly blocks sodium potassium chloride transporter, which causes the entire corticomedullary gradient to collapse, thereby preventing water reabsorption and triggering profound diuresis. That means this drug furosemide will cause increased urination, and it will prevent the reabsorption of sodium, potassium, and chloride. Finally, let us consider two high-yield exam pearls. It is important to note that juxtamedullary nephrons possess significantly longer loops of Henle that plunge much deeper into the inner medulla, enabling the production of highly concentrated urine. Furthermore, we must not overlook the role of urea, which contributes up to 50% of inner medullary osmolarity, as it is actively recycled via UTA1 and UTA3 transporters. Some people might get a bit frustrated by looking at the names of the co-transporters and transporters. Don't be too much deep into the names of the transporters, but you need to have the concept how the multiplication is occurring by different transporters, and that will get you through the exam seamlessly. Turning to slide 8, let us examine countercurrent exchange, which is specialized mechanism attributed to vasa recta. As we have already mentioned, we're not showing any canvas phase for this because we correlated both the countercurrent multiplier and countercurrent exchange in the same canvas phase. By definition, the vasa recta consists of a network of hairpin capillaries. Remember, the capillaries were like this hairpin. There was a descending portion and an ascending portion and a bend of hairpin capillary. That is the vasa recta we're talking about here again. And this vasa recta supply blood to the renal medulla. Unlike the loop of Henle, these vessels function purely as a passive exchanger, meaning their primary physiological role is to preserve the medullary osmotic gradient rather than generate it. To trace the mechanism and sequence of this exchange, we'll look first at the descending vasa recta. We mean this part. As blood flows down into the hypertonic medulla, the vessel loses water to the interstitium and simultaneously gains sodium chloride and urea, allowing the internal capillary fluid to equilibrate with the surrounding medullary tissue. So if we want to draw it again, this descending portion, it will lose water and it will gain sodium chloride and urea. Conversely, as blood flows upward through the ascending portion, this is the ascending vasa recta, the process will reverse. The vessel now will gain water back from the interstitium. That means water will get inside and it will lose sodium chloride and urea. The net result of this highly efficient loop is that absorbed solute and water are safely returned to the cortex, ensuring that hypertonic medullary gradient is continuously maintained. To clearly contrast these regional mechanisms, let us review the structural summary provided in the table. The descending loop of Henle acts as an active multiplier because it is highly permeable to water but impermeable to sodium chloride. Following this, the thick ascending loop functions as an active multiplier pump, aggressively driving sodium chloride out into the interstitium while remaining completely impermeable to water. In sharp contrast to both loop segments, both the descending vasa recta and the ascending vasa recta operate simply as passive exchangers, and both are freely permeable to both water and solutes. For the IMAT exam, there is one critical trap you must look out for. Always remember that the vasa recta does not create the gradient. Its sole responsibility is to prevent the washout of accumulated solute. As a final exam pearl to ensure coherence, a slow medullary blood flow is absolutely critical to this entire process. If the blood flow through these capillaries were to become too fast, it would inevitably wash out the medullary gradient, entirely disrupting the kidney's ability to concentrate urine. These traps and pearls are what you need to memorize and produce from your memory in the exam. Don't worry about the nitty-gritties of the loops and vasa rectas. We have now built the medullary gradient. We have a cortex at 300 milliosmoles per kilogram and an inner medullary tip at 1200 milliosmoles per kilogram. We also have a collecting duct. Let's draw the nephron again to get a clear picture, this is the Bowman capsule. Then we have the proximal convoluted tubule. Then we have the descending loop of Henle, the hairpin bend. Then the ascending one first will be thin, then it will become thick. Then we will have descending convoluted tubule, finally leading us to collecting duct. Usually we consider this upper portion to be of cortex and the lower portion to be of medulla. What I have said, let me recap again. We have a cortex at 300 milliosmoles per kilogram, which is almost equal to plasma, and an inner medullary tip, this area. This is 1200 milliosmoles per kilogram. We also have a collecting duct, let's write CD, which is descending through this gradient. But whether the body actually uses this gradient to concentrate urine depends entirely on a single small peptide hormone known as AD or antidiuretic hormone. And to decode the precise physiological controls of water conservation, we must map out the entire neuroendocrine cascade of antidiuretic hormone and urine concentration completely from scratch on our conceptual canvas. We locate the origin of this master fluid pathway within the central brain structure. Specifically, we position the specialized supraoptic and paraventricular nuclei nested within the hypothalamus as the exclusive sites where antidiuretic hormone, also known as vasopressin, is synthesized. Once this ADH is produced, this peptide hormone travels down the exoplasmic transport pathway to be stored within the terminal endings of the posterior pituitary gland. This is the pituitary gland, and this gland has two parts. One is anterior portion and the other one is posterior portion. And this ADH is stored within the terminal endings of the posterior pituitary gland, where it waits for a definitive physical or chemical release trigger. And we track this trigger mechanism by focusing on the body fluid parameter. We place specialized hypothalamic osmoreceptors alongside systemic vascular baroreceptors on our canvas. This infographic outlines the cellular mechanism of antidiuretic hormone. It also demonstrates how the water is regulated in the kidneys. We will divide the process into three distinct phases. First, we already have said these are the triggers. The first trigger is high osmolarity. This high osmolarity is detected when body fluids become too concentrated or salty, causing cells to lose water and shrink. This cell represents a shriveled cell. This word will have an L, and the spelling would be actually as HRIVELED, which means a dehydrated cell. And the low volume will be detected by the stretch receptors in the blood vessel, which are known as baroreceptors. Both of these trigger the hypothalamus to release ADH. First, hypothalamus will be triggered by both of these events, and hypothalamus will release ADH to the pituitary gland, and it will eventually come to the bloodstream. Once the ADH reaches the kidney cells, specifically the kidney ducts, remember the nephron structure, the Bowman capsule, then proximal convoluted tubule, then descending portion of loop of Henle, ascending one, collecting duct here. So the ADH function mainly happens in collecting duct. Let's draw some cells over here. 1 cell, 2, 3. Let's magnify them a bit more. ADH will come over here, and ADH will bind to the receptors present on the cell. This receptor binding will activate a protein called GS or G stimulatory protein, which will stimulate the production of cyclic AMP, and this cyclic AMP activates protein kinase A, and this whole chemical cascade triggers the cell to insert aquaporin 2 water channels. Let's draw one over here, which will be inserted in the luminal site of the cell, and through this channel water can now freely pass through the channel back to the bloodstream. So let me repeat again. The ADH hormone will come and bind to the receptors present on the cells of the collecting duct, which is called V2 receptor. V stands for vasopressin. After ADH binds to the V2 receptor of the cell membrane, it activates a chemical cascade demonstrated over here, which finally causes the opening or insertion of aquaporin 2 channels. And these aquaporin 2 channels will pull the water first inside the cells of the collecting duct, and finally, through some other channels, the water will cross the basolateral side and will come to the bloodstream, and thus the water will be reabsorbed. When ADH levels are elevated, maximum water is reabsorbed. This leaves very little water in the urine, resulting in dark, highly concentrated urine. If ADH is low, or if the kidneys are resistant to it, which is seen in a condition known as diabetes insipidus, DI, water channels are not inserted. Water cannot be reabsorbed, resulting in clear, highly diluted urine and excessive water loss. As we finish tracking this intracellular cascade from the shriveling hypothalamic cells down to the fusion of aquaporin vesicles. It started over here, and the fusion of aquaporin happened in the renal cortex and medulla. And to be more precise, in the cells of collecting duct, we see how a single endocrine messenger alters the physical permeability of a whole epithelial zone. This precise transition from chemical signaling to physical fluid shifts ensures that blood osmolarity is tightly governed, which brings us directly to our structured slide to review the core parameters, diagnostic criteria, and some other states. Now, shifting our focus to slide 9, let us delve into the regulation of antidiuretic hormone. And this antidiuretic hormone is commonly referred to as vasopressin. By definition, this vital hormone is synthesized within the hypothalamus, specifically by the supraoptic and paraventricular nuclei, before it is transported down to the pituitary stalk and ultimately released into the bloodstream from the posterior pituitary. To trace the cellular mechanism and sequence of action, we must look first at the physiological triggers. The primary stimulus for release is either an increase in plasma osmolarity or a critical drop in blood volume and pressure. Once circulating in the blood, ADH binds specifically to the G protein coupled receptors, which are known as V2 receptors, and these V2 receptors are located on the basolateral surface of the collecting duct principal cells. This binding activates the stimulatory G protein subunit, which subsequently stimulates adenylyl cyclase to generate cyclic AMP or CAMP, triggering the activation of protein kinase A. This activated pKa directly mediates the exocytosis and insertion of aquaporin 2 water channels into the apical membrane facing the tubular lumen. Consequently, water flows out of the collecting duct down its osmotic gradient into the hypertonic medullary interstitium, leaving behind highly concentrated urine. Under conditions of high ADH, this pathway ensures the excretion of a small volume of highly concentrated urine, reaching up to 1200 milliosmoles per kilogram. Conversely, a state of low ADH prevents this water reabsorption, resulting in the excretion of a large volume of highly dilute urine as low as 50 milliosmol per kilogram. When preparing for the IMAT, there are 3 distinct traps you must look out for regarding pathology and site of action. First, always remember that ADH acts physiologically on the collecting duct and it doesn't target the loop of Henle. Second, be prepared to distinguish nephrogenic diabetes insipidus, which is characterized by normal or high circulating ADH levels, but a complete failure of the collecting duct to respond to this hormone. Third, contrast this with central diabetes insipidus, a condition marked by insufficient or low ADH production that inevitably leads to profound polyuria. Polyuria means increased urination. As a final high-yield example to ensure maximum coherence, you must differentiate between the types of aquaporin channels. Aquaporin 1 channels, which are situated in the proximal convoluted tubule and the descending loop of Henle, are entirely constitutive and always open. In sharp contrast, aquaporin 2 channels in the collecting duct are always strictly ADH regulated and depend entirely on hormonal signaling to permit water reabsorption. If ADH is the kidney's water manager, then aldosterone and RAAS system are its sodium and blood pressure manager. This RAAS stands for renin, angiotensin, aldosterone. and as water follows sodium osmotically. Controlling sodium is ultimately the primary mechanism for controlling blood volume and therefore BP or blood pressure. And this RAAS is one of the most clinically important hormonal cascades in all of medicine, and the IMAT will test it repeatedly because it integrates the kidney, the liver, the lung, and the adrenal glands into a single coherent pressure sensing system. To master the dynamic feedback loops of the renin-angiotensin-aldosterone system, we must map out the entire biochemical and vascular cascade from first principles on our conceptual canvas. We locate the start of this pressure regulation network within the cortex of the kidney and the juxtaglomerular apparatus. First, let us draw a Bowman capsule, then draw glomerulus. The part entering is called afferent arteriole. Then there is a tuft and an arteriole comes out, and that is called efferent arteriole. We position the smooth muscle juxtaglomerular cells or JG cells within the walls of the afferent arteriole, and these JG cells act as primary pressure sensors. To master the dynamic feedback loops of the renin-angiotensin-aldosterone system, we must map out the entire biochemical and vascular cascade from first principles on our conceptual canvas. We locate the start of this pressure regulation network within the cortex of the kidney at the juxtaglomerular apparatus. We position the smooth muscle juxtaglomerular cells or JG cells within the walls of the afferent arteriole to act as a primary pressure sensor. Let's visualize what I have said. Imagine this is Bowman capsule. We bring in an arteriole, then there's a tuft, then an arteriole comes out. So this whole structure is glomerulus, and the arteriole that brings blood is called afferent arteriole, and the exiting arteriole is called efferent arteriole. The JG cells are situated within the walls of the afferent arterioles, and these JG cells act as primary pressure sensors. When the body suffers from dehydration, hemorrhage, or heart failure, a triple trigger occurs to prompt a renin release. The first trigger is drop in renal perfusion, and this drop in renal perfusion pressure is detected directly by the JG cells. Then the second trigger, low sodium delivery or low sodium chloride delivery, is sensed by the adjacent macula densa cells in the distal tubule. And there's another trigger, that is active sympathetic stimulation, and this is detected via a receptor called beta-1 adrenergic receptor. Upon activation, the JG cells we were talking about act as an enzyme catalyst, secreting renin directly into the bloodstream. Now we trace the biochemical transformation across the systemic circulation. We map the liver contribution here. The hepatocytes, that means the cells of the liver, continuously synthesize and secrete the large precursor protein called angiotensinogen into the plasma. The circulating renin cleaves this angiotensinogen into inactive 10 amino acid peptide known as angiotensin 1. As angiotensin 1 flows through the pulmonary capillaries of the lungs, it will encounter angiotensin converting enzyme or ACE, which is bound tightly to the endothelial cellular border. ACE will rapidly flip angiotensin 1 into fully active 8 amino acid peptide hormone called angiotensin 2. We position angiotensin 2 as the multitasking vasoconstrictor, executing 4 profound systemic and renal adjustments. First, we map selective arteriolar constriction, where angiotensin 2 powerfully constricts the efferent arteriole more than the afferent arteriole, maintaining a stable glomerular filtration rate, even when systemic blood pressure is critically low. Second, we locate direct sodium reabsorption. For that, we have drawn a portion of proximal convoluted tubule or PCT. In this proximal convoluted tubule, there will be receptors for angiotensin 2, and after binding to the receptor, it will stimulate the apical sodium hydrogen exchanger isoform 3 or NHE3. You don't need to remember the name. When this NHE3 is stimulated, it will increase sodium uptake at the tubular level. Third, it drives hypothalamic activation in the brain, stimulating the hypothalamus. This is a zoomed in section. You see hypothalamus. When hypothalamus will get stimulated, it will release ADH. But after releasing the ADH is stored in the posterior pituitary. And finally from there, the ADH gets secreted. And when ADH is secreted, it will trigger a powerful thirst signal to drive further water and volume retention. Fourth, it travels through the bloodstream to provide adrenal stimulation. We locate the target for adrenal stimulation within the outer layer of the adrenal cortex. First of all, this is kidney, and above each of the kidney we have adrenal gland, one over here and the yellow one over here. Both of them are yellow, one is right and one is left. And just like kidney, adrenal gland also has cortex and medulla. And the adrenal cortex part has so many layers, and out of the layers, one is called zona glomerulosa. And when aldosterone comes and binds to the zona glomerulosa layer, the zona glomerulosa synthesizes and releases a hormone called aldosterone. This aldosterone is highly lipid soluble. It diffuses completely free across target cell membranes, and we track its path to the final target segment, which are the principal cells of the late distal convoluted tubule and collecting duct. Aldosterone will enter into the cell and it will bind to intracellular receptors, which will cause synthesis of key transport proteins, and we position them across the cellular border. First one is epithelial sodium channel on the apical membrane, then ROMK or renal outer medullary potassium channel on the apical membrane as well, alongside sodium potassium ATPase on the basolateral side. This molecular remodeling forces rapid sodium influx from the lumen into the cell, which is backed by basolateral engine, and it simultaneously drives potassium and hydrogen ions out to the lumen, culminating in an overall net result where sodium and water are retained within the bloodstream while potassium and hydrogen are lost in urine. As we finish defining this sequential endocrine loop from the juxtaglomerular cells of the kidney to the collecting duct, we see how a physical drop in blood pressure triggers a massive biochemical transformation. The mechanical adjustment of vascular diameters work in perfect harmony with these cellular modifications of distal convoluted tubule and collecting duct to rescue systemic volume, which brings us directly to our structural slide, where we will review the core homeostatic pathways along with some traps and pearls. Aldosterone and renal renin-angiotensin-aldosterone system serve as a vital endocrine cascade, regulating blood pressure and fluid balance. To trace the mechanism and sequence of this RAAS pathway, we must begin with a state of low perfusion. That means less blood flow in the kidney, or low sodium chloride concentration at the macula densa, this is also name of cell, or increased sympathetic nervous stimulation. These physiological signals trigger the juxtaglomerular cells to secrete renin into the bloodstream. Once released, renin acts enzymatically on angiotensinogen, which is synthesized by the liver to convert it into angiotensin 1. As angiotensin 1 passes through the pulmonary capillaries, it encounters angiotensin converting enzyme or ACE, which cleaves it into highly active peptide angiotensin 2. Angiotensin 2 subsequently coordinates multiple systemic responses. It induces efferent arteriolar constriction in the kidney, stimulates the release of both aldosterone and antidiuretic hormone, triggers the brain's thirst center, and upregulates the sodium hydrogen exchanger isoform 3 or NHE3 within the proximal convoluted tubule. To understand the specific role of aldosterone, we must examine distinct cellular properties and sites of action. Aldosterone is a lipophilic steroid hormone synthesized and secreted from the zona glomerulosa of the adrenal cortex. The primary stimuli driving its secretion are circulating angiotensin II and hyperkalemia. That means increased potassium in blood. Upon release, aldosterone travels to its primary target site, acting on the principal cells of the distal convoluted tubule and the collecting duct. Its intracellular signaling cascade results in the direct upregulation of three key transport mechanisms on the principal cell membrane. First one is epithelial sodium channel, or ENAC, the renal medullary potassium channel, or ROMK, and the basolateral sodium-potassium ATPase pump. The ultimate physiological result of this coordinated upregulation is that sodium and water are retained by the body, while potassium and hydrogen ions are actively lost in the urine. For IMAT, there are three critical traps that you must look out for regarding triggers and drug interactions. First trap, always remember that high sodium does not directly stimulate aldosterone secretion. The hormone responds to volume depletion via angiotensin II and high potassium levels. Second, recognize that a state of hyperaldosteronism, that means increased aldosterone, mechanically drives hypotension due to volume expansion, accompanied by profound hypokalemia. Third, understand that administering ACE inhibitors, a drug which will inhibit ACE, causes less angiotensin II production, which downregulates aldosterone release, meaning potassium is retained by the kidney and introduces a significant hyperkalemia risk. As a final high-yield exam pearl to ensure maximum coherence on your test, you must be familiar with a pharmaceutical agent called spironolactone. Spironolactone operates explicitly as an aldosterone antagonist. By blocking the hormone's receptor, it halts the expression of epithelial sodium channel and ROMK channel, functioning clinically as a potassium-sparing diuretic that promotes fluid clearance without draining vital potassium reserves. Potassium-sparing means it doesn't excrete potassium in the urine. And when we say antagonist, it means it works against aldosterone. Every system has its counter-regulator, and the physiological counterpart to the RAAS is atrial natriuretic peptide, or ANP. If RAAS is body's alarm system for low blood pressure and low volume, ANP is the pressure relief valve for when blood pressure and volume are too high. To master the precise homeostatic functions of the kidney counter-regulators, we will look at ANP first. Then we will also look at the endocrine pathways of parathyroid hormone. Following, ANP has its origin within the muscular walls of the cardiac atria, where specialized atrial cardiomyocytes serve as volume sensors. When an excess of blood volume enters the heart chamber, it induces a physical atrial stretch. This mechanical expansion triggers these cardiomyocytes to rapidly synthesize and release ANP directly into the systemic circulation, where it travels to the kidneys to act as a direct physiological RAAS antagonist. We trace the mechanical actions of ANP as it enters the renal corpuscle within the cortex. Remember renal corpuscle? Glomerulus and Bowman capsule together form renal corpuscle. That's an IMAT question. When this ANP enters the renal corpuscle within the cortex, something interesting happens. Let's draw the first part of the nephron. This is Bowman capsule, yet again, and we put afferent arteriole, then efferent arteriole. ANP actively drives afferent arteriole dilation while simultaneously forcing efferent arteriole constriction. By widening the inflow channel and narrowing the outflow pipe, a massive surge of blood is backed up within the capillary loops, increasing glomerular hydrostatic pressure and successfully driving the filtration rate, or GFR, upward to force more fluid into Bowman space. Moving downstream, ANP executes a triple inhibition of retentive hormones, traveling to specific tissue sites to completely shut down the secretion of renin, aldosterone, and anti-diuretic hormone. Furthermore, inside the medullary collecting duct, let's erase these names to extend our nephron. Our target is to show you the medullary collecting duct. So let's say proximal convoluted tubule, then loop of Henle, then distal convoluted tubule, and then collecting duct. Collecting duct has cortical portion. Let's say this is the cortical one and this is the medullary one. So what ANP does is ANP binds to the receptors over here and it inhibits sodium reabsorption. Because the retentive hormones are paralyzed and the collecting duct transport channels are blocked, massive quantities of sodium and water are left trapped inside the lumen, culminating in a powerful result of natriuresis and diuresis that actively reduces overall blood volume and drops blood pressure back to normal. Now we construct the second independent regulatory system on our canvas, which is parathyroid hormone or PTH, which operates as the primary calcium mobilizer. We locate its source within the neck, where the parathyroid glands release PTH into the bloodstream in response to a low ionized plasma calcium signal known as hypocalcemia. PTH circulates to the kidney, where it targets two completely separate tubule zones to execute an elegant dual manipulation of mineral transport designed to restore calcium balance while preventing dangerous mineral precipitation in the blood. First, we trace PTH down to the distal convoluted tubule. We target this DCT for regulated calcium retention. On the apical membrane of these DCT epithelial cells, PTH upregulates the expression and open state probability of transient receptor potential vanillate member 5 channel or TRPV5. You can forget it, it's a bit advanced term, but I'm introducing it for comprehension. So when this TRPV5 is upregulated, it actively pulls calcium out of the lumen and returns it to the bloodstream to elevate plasma calcium levels. Let's say this is DCT, only one portion, and we have cells over here, and on the apical side we have TRPV5 transporter. This pulls calcium from the lumen and puts it into the bloodstream, and this elevates plasma calcium level. Second, we trace PTH back to the proximal convoluted tubule. Let's say we draw proximal convoluted tubule here, only a portion, and here the parathyroid hormone executes an opposite transport command to cause profound phosphaturia on this proximal brush border. Let's say this is a cell and we will have brush border here. PTH will bind and it will directly inhibit a channel called sodium phosphate I or sodium inorganic phosphate cotransporter. By blocking the proteins or the transporters, phosphate transport will be halted, and phosphate is intentionally lost in the urine. That means phosphate cannot get into bloodstream anymore and they are destined to flow out as urine. And by forcing this phosphate loss while simultaneously retaining calcium, the kidney prevents the two ions from binding together and forming solid calcium phosphate mineral crystals in the bloodstream. Finally, we map the kidney function as a specialized vitamin D factory within the proximal tubule cells. PTH does one thing. PTH will directly activate the enzyme called 1-alpha hydroxylase. This active enzyme converts the inactive circulating precursor vitamin D into a fully active steroid hormone which has a mouthful name called 1,25-dihydroxycholecalciferol or simply calcitriol. This calcitriol will be secreted into blood where it travels to the intestinal mucosa. It will travel to intestine to aggressively increase gut calcium absorption, completing the triple threat mechanism to raise systemic calcium. As we finish tracking these counter-regulatory signals from the stretching chambers of the heart and the calcium sensors of the neck down to the microscopic channels of the nephron, we see how the structural layout of separate segments allows independent adjustments of blood pressure and mineral matrices. This precise coordination ensures that while one hormone reads the lumen of excess fluid volume, another independent messenger chemically alters the mineral valences downstream, which leads us directly to our structured side review to anchor these core counter-regulatory definitions, traps, and pearls. By definition, atrial natriuretic peptide, or ANP, is a polypeptide hormone synthesized and released directly by atrial cardiomyocyte. The primary physiological stimulus driving its secretion is increased atrial stretch, which acts as a direct mechanical indicator of high blood volume. Once released into the systemic circulation, ANP exerts potent renal effects by inducing afferent arteriolar dilation while simultaneously causing efferent arteriolar constriction, a coordinated hemodynamic shift that significantly increases the glomerular filtration rate. Furthermore, ANP directly downregulates fluid retention by inhibiting the secretion of renin, aldosterone, and antidiuretic hormone. At the tubular level, it explicitly blocks sodium reabsorption within the collecting duct. The cumulative physiological result of these actions is a profound state of natriuresis and diuresis, which successfully brings about a dramatic fall in both blood volume and systemic blood pressure. Turning our attention to the second regulator, parathyroid hormone, or PTH, is a peptide hormone synthesized and secreted by the parathyroid gland. This is thyroid gland, and we draw 4 parathyroid glands like dots over here. So we have 4 parathyroid glands. Nice to know. The primary homeostatic stimulus triggering the release of parathyroid hormone is a state of hypocalcemia. That means low calcium level in blood. Once active, PTH targets the distal convoluted tubule to actively stimulate calcium reabsorption through the upregulation of TRPV5 channels, effectively raising plasma calcium levels. Concurrently, it acts on the proximal convoluted tubule where phosphate reabsorption is completely inhibited, leading to a marked state of phosphaturia. Beyond direct ion transport, PTH exerts a critical endocrine effect on the kidney by upregulating the enzyme 1-alpha hydroxylase, which converts inactive 25-hydroxyvitamin D into its active form 1,2,5 or 1,25-dihydroxyvitamin D, or simply put, calcitriol. So this enzyme is very important for converting the 25-hydroxyvitamin D to its active form 1,25-dihydroxyvitamin D or calcitriol. And for IMAT, let us review the traps, and these traps are the ones that you need to remember. First one, remember that ANP functions strictly as a direct RAAS antagonist. While the RAAS pathway promotes fluid and sodium retention, ANP aggressively promotes their excretion. Second, keep in mind that PTH drives calcium and phosphate transport in entirely opposite direction, stimulating calcium retention while forcing phosphate excretion. So the bottom line is written in exam pearls. Parathyroid hormone has 3 actions. The first one being calcium retention, and it does it in the distal convoluted tubule of nephron. Then it does calcium reabsorption, and this calcium reabsorption is done via intestine. And when parathyroid hormone activates vitamin D does the job of calcium reabsorption from intestine. Then when it's required, parathyroid hormone breaks down the bone and releases calcium into the blood. That's why we can say that parathyroid hormone causes hypercalcemia, or it can bring back the value of calcium level to the normal range in blood. Every metabolic process in the body produces acid. Aerobic respiration releases carbon dioxide, which forms carbonic acid. Anaerobic respiration produces lactic acid. Amino acid catabolism, that means when we break down amino acid, generates sulfuric acid and phosphoric acid. Fat metabolism yields keto acid. Despite this relentless acid production, blood pH stays within the very narrow window of 7.35 to 7.45. And 3 systems maintain this stability. First one, chemical buffers in the blood acting instantly, respiratory regulation, acting in minutes, and the renal regulation acting over hours to days. Respiratory regulation is already covered in the respiratory system lecture, so we will cover renal regulation and chemical buffers of blood in this section. And we will start with renal regulation. All right, now let's break down exactly how the proximal convoluted tubule handles the phase 1 or bicarbonate reclamation. Let's trace it out step by step. First, look at the apical membrane. What I have done is I have shown a segment of proximal convoluted tubule and only one cell. As I need to annotate so many things, I have zoomed in, magnified this cell, and I have brought the same cell over here. This side represents apical membrane and this side represents basolateral membrane. So for reference, write them down. So actually, we will pivot and see the magnified cell, and we will try to draw everything around this magnified image. Okay, let's first look at the apical membrane. You see the brush borders facing the lumen. And here sits our main engine, which is NHE3, this guy. This is a secondary active transport antipode. It harnesses the down gradient movement of sodium into the cell to pump a hydrogen ion out into the tubular lumen. And with black color code, I try to represent hydrogen ATPase pump, and make a note here. This one is primary active transport. It spends ATP directly to shove protons out their concentration gradient. Both mechanisms serve one purpose: load the lumen with proton. Now look what happens in the urine. That secreted proton meets our filtered bicarbonate. So we write HCO3 for it. And both of them together will form carbonic acid, H2CO3. Now you might think why this is happening. Because bicarbonate wants to get inside the cell, but it cannot get inside due to its negative charge. So it masks itself and it forms a bond with proton to make carbonic acid. And when carbonic acid is formed, this carbonic acid is broken down by Ca4 or carbonic anhydrase 4. And this carbonic acid is then converted into carbon dioxide and water, H2O. Because carbon dioxide and water are small non-polar, they glide straight through the membrane into the cell. So let's say we put carbon dioxide inside and water inside as well. Once inside the cytoplasm of the cell, we reverse the entire process. Here, dissolved in the cytosol, carbonic anhydrase 2 quickly transforms the water and carbon dioxide to become carbonic acid again. And this carbonic acid spontaneously breaks down into bicarbonate, HCO3- and proton. Be careful with this proton that is generated now. This proton doesn't leave the cell toward the blood. The proton will get recycled right back to the apical membrane to go handle the next incoming bicarbonate ion, and it will use the same pump, NHE3, to get out. It is becoming a bit clumsy, that's why we're not drawing that anymore. But what needs to be drawn is the basolateral exit of bicarbonate. And for basolateral exit, bicarbonate will use NBC1 or sodium bicarbonate cotransporter 1 to go to the bloodstream. And thus the pH is maintained. Now to recap, if a question asks about Ca4, write down extracellular or brush border. If the question asks about Ca2, write down intracellular or cytoplasm. And remember, bicarbonate is never reabsorbed directly. It is a molecular magic, changes into gas to slip inside and reforms on the other side. And I have mentioned the reason. As bicarbonate has charge, it cannot cross the lipid bilayer membrane by itself. Now that we understand how the proximal tubule reclaims filtered bicarbonate, we have to look at how the kidney actually gets rid of net daily acid loads. Which brings us to renal buffer systems, mechanism for acid excretion. Remember from our earlier discussion, the kidney cannot push the luminal pH down below that absolute wall of 4.4. If we left protons free in the urine, we would hit that limit instantly and stop excreting acid. To beat this, the intercalated cells use two major chemical buffer traps. Let's map out how they work under IMAT scrutiny. Before looking at the buffers, look at the cell engine itself. Inside the type A intercalated cell, carbonic anhydrase hydrates carbon dioxide and water into carbonic acid, which splits into proton and bicarbonate ion. On the apical membrane facing the lumen, the hydrogen ATPase pump actively handles primary active transport, pushing the free protons out to the lumen. And meanwhile, look at the basolateral membrane facing the blood peritubular capillary. The regenerated bicarbonate exits into our blood via chloride bicarbonate exchanger antiport. This is how the cell continuously supplies new base to the body while tossing acid into the urine. Now let's look at our first system, the phosphate buffer. Monohydrogen phosphate is filtered at the glomerulus and runs down the tubule. When it encounters that freshly secreted proton from our ATPase pump, they combine. They turn into dihydrogen phosphate, and we call this titratable acidity. Look at the high yield node on the far left. It has a pKa of 6.8, which falls perfectly within the functional range of urine pH. Once dihydrogen phosphate is formed, it is trapped and safely excreted in the urine. Protons masked, problem solved. Now for the heavy lifter during chronic or severe acidosis, the ammonia buffer system. Unlike phosphate, ammonia is dynamically synthesized by proximal tubule cells directly from amino acid glutamine. Look at its properties here. Ammonia is lipid soluble. This means it effortlessly crosses the cell membrane lipid bilayer by simple diffusion down into the lumen. You might notice that there was infographic of ammonia buffer system over here, but we're not going to use the diagram because it has some flaws in it. We're going to discard it, but the infographic that we use to explain phosphate buffers is correct, so we will lean on that. To explain the ammonia buffer system, we're going to look at the hand-drawn diagram. This is a segment of proximal convoluted tubule, and this one is a proximal tubular cell. The proximal tubule cells directly from the amino acid glutamine synthesizes ammonia, and this ammonia is lipid soluble. This means it effortlessly crosses the cell membrane lipid bilayer by simple diffusion down into the lumen. So in lumen we have ammonia now, but the moment ammonia enters the lumen and meets a secreted proton, we know where proton comes from. This reaction makes ammonium ion. You see, ammonium is highly charged and water soluble ion, and that's why the lipid bilayer becomes an absolute permeability barrier to it. It cannot diffuse back into the cell. And this is the definition of charged trapping or ionic trapping. It locks the acid into the lumen permanently, allowing the body to dump vast quantities of protons without violating that 4.4 pH wall. Moving on to slide 12, we will look at the exam traps and pearls for renal acid-base regulation, and we will solidify what we have seen in our conceptual canvas. And starting with core definitions, when it comes to managing the body's pH, the kidneys handle long-term regulation over hours to days, whereas the lungs manage short-term regulation within minutes via carbon dioxide excretion. Looking at the precise mechanism and sequence, the proximal convoluted tubule or PCT handles bicarbonate reclamation, which is distinct from NAT acid excretion. In this specific pathway, hydrogen ions are secreted into the lumen via NHE3 and combined with filtered bicarbonate to form carbonic acid, which is broken down into carbon dioxide and water by carbonic anhydrase 4. If you can remember the whole sequence, at least you remember that the enzyme responsible is carbonic anhydrase. Then the carbon dioxide that was produced enters the cell, where intracellular carbonic anhydrase 2 converts it back into carbonic acid, which dissociates into hydrogen ion and a bicarbonate ion. Next, the bicarbonate exits the basolateral membrane into the blood, while the hydrogen ion is recycled to the lumen. down the nephron in the alpha intercalated cells of the collecting duct, hydrogen ATPase and hydrogen potassium ATPase secrete hydrogen ions to add completely new bicarbonate to the blood. To prevent the environment from becoming too acidic, urine buffers like phosphate acting as titratable acid and ammonium excretion step in to catch those ions. This sequence brings us straight to the high-yield exam traps you need to watch out for. First, always remember that PCT, bicarbonate reabsorption, is reclamation, not net acid excretion. Second, keep in mind that minimum urinary pH is approximately 4.4, meaning ammonium is strictly required to handle large acid loads. Third, note that the drug acetazolamide inhibits carbonic anhydrase, causing bicarbonate to be lost in the urine, which leads to alkaline urine despite an underlying metabolic acidosis. Some people might get intimidated by the amount of information I share, but don't worry about too much information. I just want to make sure that you understand the underlying concept and you can produce from your memory whenever a question asks you to do that. You can omit the name of the transporters, but you need to know what type of mechanism occurs. Finally, we close out the slide with two examples. In a state of metabolic acidosis, the kidney actively compensates with more hydrogen secretion, more bicarbonate generation, and more ammonium production. By contrast, in respiratory acidosis, the renal compensation relies on increased bicarbonate reabsorption, which is a slow process taking days to complete. One of the most reliable IMAT question formats is the composition comparison. Given a table of plasma, filtrate, and urine values, identify which substance is present in which compartment at what concentration and explain why. To answer these questions correctly, you need to understand the relationship between the three compartments as a flow-through system with specific handling at each stage. To master renal fluid dynamics, we must track the entire molecular journey from plasma to urine by constructing the composition changes completely from scratch on our conceptual canvas. We locate our starting point inside the glomerular capillary lumen. This is the lumen, this is the afferent arteriole, and this is the efferent arteriole of glomerulus. Inside the lumen, we analyze the incoming systemic plasma. Systemic plasma means the plasma of the body blood. This blood fluid minus its cellular components contains stable homeostatic levels of glucose and electrolytes alongside an exceptionally high concentration of protein, measured at a baseline albumin value of 7 grams per deciliter. Now the fluid encounters the filtration barrier. This whole part is the barrier. This triple layered this triple layered sieve blocks large proteins and intact blood cells from leaving the capillary. If these components appear in the subsequent segments, it indicates severe physical damage to the glomerular basement membrane. Solutes smaller than 70 kilodalton pass completely free, creating the filtrate within Bowman space. This is the Bowman space, or sometimes called Bowman capsule. To be more precise, this whole structure is the Bowman capsule, and the space between two layers, this space is called the Bowman space. And this Bowman space acts as a protein-free mirror of the plasma. Within this ultrafiltrate, protein is entirely absent, but glucose matches plasma concentrations exactly at approximately 5 millimoles per liter. Similarly, urea sits at 2.5 to 7 millimoles per liter. Creatinine is at 90 micromoles per liter. Sodium ions match at 140 millimoles per liter, and bicarbonate matches plasma at 24 millimoles per liter. The pH shifts slightly down from the systemic plasma window of 7.35 to 7.45, stabilizing at an initial filtrate pH of approximately 7.35. As the ultrafiltrate leaves Bowman space, it enters the specialized tubule sections, the first one being proximal convoluted tubule. And here, the specialized structural hierarchies altered the chemical layout. First, the fluid winds through the proximal convoluted tubule here, which serves as the massive recovery zone. We map glucose threshold given by SGLT2 transporters over here. Glucose is 100% reabsorbed below a transport maximum corresponding to a plasma threshold of 180 milligrams per deciliter. You need to remember this value. You can forget all this. I have just added these values for context. Moving down into the loops and entering the final processed end product phase, the fluid undergoes extensive concentration and fine-tuning. We observe a dramatic divergence in how the kidney concentrates urea versus creatinine within the final urine. As the fluid flows through the medullary collecting duct, we locate specialized transporters that actively recycle a large portion of the filtered urea back into the medullary interstitium to maintain the hyperosmotic gradient. We have already discussed, that's why I'm just giving a light touch of all the concepts again. In sharp contrast, we position creatinine as an unrecycled waste molecule that is only minimally secreted, meaning its clearance provides a pure proxy for overall filtration. That means you can check the filtration situation by measuring creatinine. That's why sometimes it's called the gold standard test or kidney function test. Because some urea is recycled into the tissue while creatinine remains entirely trapped within the lumen, creatinine is concentrated approximately 100-fold in the final urine compared to plasma, whereas urea is only concentrated 20-fold. Electrolyte concentrations become highly variable. Sodium levels fluctuate based on aldosterone-dependent ENAC tuning, and bicarbonate drops to exceptionally low levels or is completely absent during states of systemic acidosis. Finally, the hydrogen ion pumps create a highly variable final urine pH window, ranging from a high acidic 4.4 to an alkaline 8.0 depending on the body homeostatic requirements. Turning the page to slide 13 again, we transition directly into the quantitative composition comparison between plasma, filtrate in Bowman's space, and the final urine to understand renal handling. Beginning with our key features table, we look first at protein, specifically albumin, because this is the main plasma protein responsible for generating oncotic pressure, sits at 7 gram per deciliter in the plasma, but it is completely absent in the initial filtrate and remains absent in the final urine unless glomerular filtration barrier damage has occurred. Next, glucose presents at approximately 5 millimoles per liter in the plasma and is freely filtered into Bowman's space at that exact same concentration of about 5 millimole per liter. Yet it is completely absent in the final urine because it is fully reabsorbed. Moving to urea, it maintains a low plasma level between 2.5 and 7 millimoles per liter, matches this level identically in the initial filtrate, but becomes heavily concentrated down the nephron to arrive at roughly 20 times its plasma concentration in the final urine. Creatinine allows a similar trajectory, starting at a normal plasma baseline of about 90 micromoles per liter, remaining the same in the filtrate but becoming massively concentrated up to 100-fold by the time it reaches the final urine. For sodium, the concentration is strictly regulated at approximately 140 millimoles per liter in both the plasma and the filtrate, but turns highly variable in the final urine under the direct influence of aldosterone. Bicarbonate shows a parallel trend, holding at a steady 24 millimoles per liter in the plasma and filtrate, but dropping to low levels in the final urine due to extensive reabsorption and becoming entirely absent during the states of systemic acidosis. Finally, hydrogen ion concentrations are best viewed through pH. Shifting from a tightly controlled plasma pH of 7.35 to 7.45 down to a slightly more acidic filtrate pH of approximately 7.35 before opening up to a highly variable final urinary pH range of 4.4 to 8. This quantitative baseline brings us straight into our critical exam traps. The first high-yield trap to remember is that finding glucose in the filtrate does not mean you will find glucose in urine, because it is entirely reabsorbed at the proximal convoluted tubule as long as blood glucose remains below its transport maximum or TM. Second, you must remember that unlike glucose variations, finding protein in the urine is always pathological, indicative of filtration barrier compromise. We close out this comparison with an essential example regarding clearance markers. Because the kidney concentrates urea roughly 20 folds, creatinine serves as a far superior marker for calculating the clinical glomerular filtration rate due to lack of absorption of creatinine and concentrating 100-fold due to lack of reabsorption. Now we move to the topic of diuretics. Diuretics are moderately important for the IMAT. While they do not appear on every exam, you are frequently tested on their fundamental pharmacology, specifically how they affect kidney function and blood pressure, making them a key topic within the syllabus's medical and biological sections. To master the precise pharmacology of diuretics, we must build the entire molecular mapping of drug mechanisms directly on our conceptual canvas of the nephron. We locate these separate pharmacological interventions distributed sequentially from the cortical headwaters down to the medullary collecting duct. Each class of medication acts as a specific molecular key designed to bind a distinct transport protein or enzyme, altering the physical permeability and electrical gradients to drive fluid excretion. We begin our journey in the renal cortex by mapping out the first site of action within the proximal convoluted tubule. Our point of interest is this portion, PCT. Here, we position the carbonic anhydrase inhibitors, exemplified by the generic chemical entity acetazolamide. Acetazolamide targets and blocks the enzyme carbonic anhydrase both on the brush border membrane and inside the cell cytoplasm. By freezing this enzymatic cycle, it prevents the conversion of luminal bicarbonate into diffusible carbon dioxide gas, halting bicarbonate reabsorption. This leaves the bicarbonate trapped inside the lumen as an osmotic solute, forcing a mild diuresis and producing an alkaline urine, while causing a systemic metabolic acidosis. Right beside it in the PCT and the descending loop, right around here, we place the osmotic diuretics, exemplified by the sugar molecule mannitol. Mannitol is an inert substance that is freely filtered across the glomerulus. That means mannitol can come from glomerulus into the tubule, but it cannot be reabsorbed by any subsequent tubule segment. As a result, it remains completely trapped within the tubular lumen throughout the entire nephron. The mannitol keeps coming here, let's say M, but it is trapped. It doesn't have any scope for reabsorption, and it exerts a powerful osmotic pull that physically holds water molecules inside the lumen and prevents water reabsorption. That means it locks in water with it, making it exceptionally useful in emergency clinical protocols to manage cerebral edema or acute glaucoma. Now we dive vertically down into the medulla to map absolute powerhouse class of diuretics, the loop diuretics, exemplified by the generic drug furosemide. We target the thick ascending limb of the loop of Henle as their exclusive site of action. Here we position furosemide binding directly and selectively on the sodium-potassium 2-chloride cotransporter or NKCC2. By blocking this active transport engine, the drug halts all the accumulation within the medullary tissue. That means this transporter was supposed to absorb sodium, then potassium and chloride. But furosemide does not allow it. As a result, it completely collapses the hyperosmotic medullary osmotic gradient. Because the tissue gradient is destroyed, water can no longer be drawn out of the collecting ducts, resulting in a massive output of dilute urine. This aggressive solute flushing induces notable side effects, including heavy potassium loss. That means potassium can be lost with urine and calcium loss into the lumen, emerging back into the cortex. That means we're going up again. The tubule transitions into distal convoluted tubule, roughly here, where we position the thiazide diuretics, exemplified by hydrochlorothiazide. Thiazides target the neutral sodium chloride co-transporter or NCC, embedded along the apical membrane of the DCT. This class acts as a first-line therapy for uncomplicated hypertension, producing a moderate diuresis. As it blocks the NCC co-transporter, it triggers a secondary potassium loss due to increased sodium and fluid delivery downstream. But it executes a unique twist on divalent cations by driving calcium retention. That means this thiazide diuretic pulls calcium out of the urine back into the blood, making it highly useful for treating calcium stone disease. Finally, we track the tubule system into its terminal adjustment zones, which are the late distal convoluted tubule and the collecting duct, to map the potassium-sparing diuretics. We divide this class into two distinct sub-mechanisms. Let's say we show all of them at collecting duct for our convenience. So the first one is direct aldosterone antagonist like spironolactone and epithelial sodium channel blockers like amiloride. We locate the target for spironolactone inside the cell nucleus. By cell, we mean the cells of the tubule or collecting duct. This spironolactone binds directly to the aldosterone receptor to prevent aldosterone binding. That means spironolactone takes the place of aldosterone and it doesn't let aldosterone bind anymore to their receptor. And this blocks the transcription and upregulation of ENAC. Let's say this is a cell. It was supposed to have two channels. Let's say one channel here, ENAC, actually here, apical membrane. Another one, let's say this one, is ROMK. When both of the channels are blocked, potassium cannot get out, and at the same time, sodium cannot be reabsorbed. And amiloride acts directly on the apical surface to physically plug those ENAC channels. That means amiloride will directly get attached with ENAC or epithelial sodium channel. By preventing aldosterone from upregulating these proteins, the sodium-potassium seesaw will be halted. And what is the net result? The net result is a mild diuresis of high clinical value, where sodium is lost in the urine, but potassium is safely retained within the blood, making these agents invaluable for preventing dangerous hypokalemia when combined with loop or thiazide diuretics. As we finish defining this pharmacological layout from enzymatic blocks of the proximal tubule to the receptor antagonism of the collecting duct, we see how every individual drug class manipulates specific ion channels. This targeted disruption of transport protein alters the electrical potential and osmotic forces throughout the tubule, which brings us directly to our structured slide to review these core diuretic definitions, traps, and diagnostic pearls. Beginning with our key features table, we first examine loop diuretics like furosemide, which act specifically on the thick ascending limb to block the NKCC2 co-transporter, collapsing the medullary osmotic gradient to trigger a large diuresis alongside significant potassium loss. Next, thiazides such as hydrochlorothiazide target the distal convoluted tubule or DCT, where they inhibit the NCC sodium chloride co-transporter to produce moderate diuresis, notable calcium retention, and concurrent potassium loss. Moving to potassium-sparing diuretics like spironolactone, these agents work at the level of DCT and collecting duct by blocking the aldosterone receptor, which promotes sodium loss while keeping potassium safely retained. Following this, carbonic anhydrase inhibitors, specifically acetazolamide, exert their effect in the proximal convoluted tubule, or PCT, by inhibiting carbonic anhydrase enzyme, which results in bicarbonate loss, mild diuresis, and systemic metabolic acidosis. Finally, osmotic diuretics like menthol function across both PCT and loop of Henle through a purely physical osmotic effect without blocking any transporter, which effectively prevents water reabsorption and makes them highly useful in managing cerebral edema. This pharmacologic breakdown brings us straight to the high-yield exam traps you need to master. First, always remember that loop diuretics cause potassium loss leading to hypokalemia, whereas spironolactone directly prevents this depletion. Second, pay close attention to calcium handling, as thiazides act to retain calcium, whereas loop diuretics cause calcium loss in the urine. We close out this sequence with 3 essential exam pearls linking specific drugs directly to our molecular targets. For your test, firmly correlate that molecular target for furosemide is the NKCC2 transporter. The target for acetazolamide is the carbonic anhydrase enzyme. And the target for spironolactone is aldosterone receptor. It's a bit advanced for many students. So most of the students, you can just remember the name of the diuretics and discard other mechanisms. But if you want to ace your exam, then I will highly recommend you also study these kinds of depths according to your capability. The kidney does not just regulate individual solutes or individual hormones in isolation. Its overarching mission is whole body fluid and electrolyte balance or homeostasis. 2 separate but interacting variables are being continuously monitored and corrected. One is plasma osmolarity and the other one is circulating blood volume. These are not the same thing and they're not always regulated in concert, which is a source of confusion that the IMAT exploits. And to master the precise mechanics of fluid balance, we must deconstruct the intricate dual network of osmoregulation and blood volume management completely from first principle. We locate this physiological control center functioning as a delicate balance. As we have said, there are 2 parameters. One is plasma osmolarity. This affects or reflects solute concentration, and circulating blood volume, which dictates the physical pressure. This distinct homeostatic system work in parallel across the human body, but they utilize entirely separate sensory cell populations and distinct hormonal effectors. We will build our foundation of this fluid scale by establishing the strict quantitative laws of body water distribution. We map the total body water, which accounts for exactly 60% of total human body weight. We slice this 60% total into 2 main compartments according to the 60-40-20 rule. First, we locate the largest fluid compartment, the intracellular fluid, containing 40% of total body weight inside the cellular border. Second, we position the remaining 20% as the extracellular fluid compartment outside the cell membranes. We further subdivide this extracellular zone into 2 subcompartments. 15% is localized within the interstitial space bathing the tissues, and the final 5% is trapped inside the vascular system as plasma volume. Now we will focus more specifically to construct the osmoregulation network, which is tightly specialized for maintaining plasma osmolarity within a narrow window of 285 to 295 milliosmoles per kilogram. We position the primary sensors, the hypothalamic osmoreceptors, within the brain tissue. We trace 2 opposite response mechanisms from these sensors. When a patient experiences a water excess, plasma osmolarity falls below 285 milliosmoles per kilogram. This drop suppresses the release of antidiuretic hormone, rendering the collecting duct entirely impermeable to water and forcing the kidney to excrete excess water as a massive volume of highly dilute urine, reaching a concentration extreme of 50 milliosmol per kilogram. Conversely, when a patient suffers a water deficit, plasma osmolarity rises above 295 milliosmoles per kilogram. This rise triggers immediate ADH release from the posterior pituitary, activating V2 receptors to insert aquaporin 2 channels into collecting duct apical membrane that we have discussed, and it causes the tissue to retain water and dilute the blood, leaving behind a small volume of highly concentrated urine reaching a concentration extreme of 1200 milliosmoles per kilogram. Thus, ADH acts as the primary hormonal effector for osmolarity. Next, we will look at the other side of our balance scale to build the blood volume and physical pressure response mechanism, which ensure adequate cardiac output. We position the primary pressure sensors, the high pressure and low pressure baroreceptors within the cardiac atria, the carotid sinuses, and the aortic arch. To get more information on this, you can watch out our circulatory system lecture, lecture 10. We trace two opposite volume responses from this vascular point. When a patient undergoes severe volume depletion, such as a major acute hemorrhage or severe dehydration, the drop in stretch triggers the primary hormonal effectors of volume, which are the renin-angiotensin-aldosterone system and ADH. Activated RAAS surges renin, angiotensin 2, and aldosterone levels to upregulate sodium and water reabsorption, producing a small volume of highly concentrated urine up to 1200 milliosmoles per kilogram to stabilize systemic pressure. Conversely, when the body experiences a volume excess, the resulting high vascular pressure induces an intensive atrial stretch. This mechanical stretch triggers the release of atrial natriuretic peptide or ANP from the heart walls, which binds to the renal tubules to promote active natriuresis and diuresis, yielding a large volume of dilute urine at 50 milliosmoles per kilogram to bring blood volume and physical pressure down. Finally, we must map out the physiological stress intersections where these two systems conflict. In states of catastrophic volume depletion like a massive hemorrhage, the body encounters a severe non-osmotic override. The vital baroreceptors sense the drop in physical pressure and completely hijack the system, sending powerful neural signals that override the osmoreceptors, forcing continuous massive ADH release, even if the plasma osmolarity is already normal or low. This prioritizing of pressure over concentration represents a vital protective compromise to maintain central perfusion. Then we have a condition called SIADH. In this condition, ADH becomes too much and it causes too much water reabsorption. As we complete this detailed mapping of the 60-40-20 rule and the parallel sensors of the heart and brain, we see how the body prioritizes survival by separating concentration from physical pressure. The mechanical override of osmoregulation during a hemorrhagic shock highlights how cellular pathways shift focus dynamically under severe physiological stress, which brings us directly to our structured slide review to anchor these core parameters, clinical traps, and standout stage. After advancing to slide 15, we start with definition. First, osmoregulation is defined as the process of maintaining plasma osmolarity within a tight homeostatic range of approximately 285 to 295 milliosmoles per kilogram. Directly linked to this balance is total body water, which accounts for roughly 60% of total body weight, further divided into an intracellular fluid compartment of 40% and an extracellular fluid compartment making up the remaining 20%. Looking at the precise homeostatic mechanisms and sequences, a state of water excess causes plasma osmolarity to fall, which suppresses antidiuretic hormone or ADH, leading to the excretion of highly dilute urine as low as 50 milliosmoles per kilogram. Conversely, a state of water deficit causes systemic osmolarity to rise, triggering the release of ADH to produce a heavily concentrated urine, reaching up to 1200 milliosmoles per kilogram. Beyond pure water shift, a state of volume depletion activates baroreceptors, which stimulate both ADH and the renin-angiotensin-aldosterone system, to drive coordinated sodium and water retention. On the flip side, a state of volume excess causes mechanical atrial stretch in the heart, releasing atrial natriuretic peptide or ANP, which promotes rapid natriuresis and diuresis to lower blood volume. This regulatory behavior brings us straight to two high-yield exam traps that frequently show up on IMAT questions. First, you must remember that osmolarity and volume are regulated completely independently, meaning that RAAS axis responds specifically to blood volume, while osmoreceptors track plasma osmolarity. Second, pay close attention to the fact that ADH can be strongly stimulated by volume depletion, even if systemic osmolarity happens to be perfectly normal or low. We close out this slide with an essential exam pearl regarding the syndrome of inappropriate ADH or SIDH. In this specific pathology, excess ADH causes marked dilutional hyponatremia despite patient maintaining a clinically normal blood volume status. We have covered a remarkable amount of ground in this lecture, and before you move to our practice questions, I want you to walk through the anchor slide for this final section the way a good teacher does before an exam. Not a list read aloud, but a rapid synthesis connecting every keyword back to the mechanism that makes it true. Think of it as the final imprint on your memory before the material set. Starting out with key features. The thick ascending limb is strictly impermeable to water and express NKCC2 co-transporter, which serves as the primary target for loop diuretic. Moving to the proximal convoluted tubule, SGLT2 handles glucose reabsorption up to a plasma threshold of approximately 180 milligrams per deciliter. Next, tracking hormone pathways. Antidiuretic hormone is released from the posterior pituitary, binding to V2 receptors to activate a cyclic AMP second messenger system that inserts aquaporin 2 channels into the collecting duct. Similarly, aldosterone is secreted from the adrenal cortex to increase ENAC and ROMK expression, driving sodium reabsorption and potassium excretion. This ties directly into the broader RAAS axis, where renin from juxtaglomerular cells converts angiotensinogen to angiotensin 1, which is cleaved by ACE in the lungs into angiotensin 2 to trigger aldosterone release. Opposing this system is atrial natriuretic peptide, released from the atria, which actively counters RAAS to promote natriuresis and diuresis. Looking at calcium homeostasis, parathyroid hormone stimulates calcium reabsorption in the distal convoluted tubule, drives phosphate excretion in the proximal convoluted tubule, and activates calcitriol. For physical mechanisms, the countercurrent system uses the loop of Henle as a multiplier and vasa recta as an exchanger to maintain the medullary gradient. This ties back to systemic homeostasis, where acid-base handling is split between lungs, which react fast within minutes, and the kidneys, which react slowly over hours to days. We close the key features with quantitative filtration values, where the net filtration pressure is calculated as 60 minus 15 minus 29 minus 0, yielding 60 millimeters of mercury, which drives a normal glomerular filtration rate of 125 milliliters per minute or 180 liters per day. This rapid-fire foundation brings us straight into 3 high-yield exam traps to avoid on test day. First, always remember that ADH acts specifically on collecting duct, not on the loop of Henle. Second, keep in mind that it is specifically juxtaglomerular nephrons with their long loops that are responsible for creating highly concentrated urine. Third, do not confuse your diuretic classes. Remember that spironolactone is potassium sparing, whereas furosemide is strictly potassium wasting. We close out this comprehensive review slide with indispensable clinical example. For autoregulation, the GFR is strictly maintained across a mean arterial pressure range of 80 to 180 millimeters of mercury, meaning that serum creatinine will rise when the GFR explicitly falls. Finally, always remember the exact rule of glomerular filtration, which states that the initial filtrate is identical to plasma minus proteins and blood cells, meaning everything else passes through at the exact same concentration. We're now moving into our dedicated question-solving session. In an exam like the IMAT, simply knowing the theory of the renal system is not enough. Solving high-quality MCQs is absolutely vital because it trains your pattern recognition and forces you to apply complex physiological concepts under strict time pressure. The IMAT heavily tests your critical thinking through multiple-choice formatting. So mastering these specific questions is what ultimately bridges the gap between basic understanding and a top-tier competitive score. Let's dive straight into the first question. Never let examiners trick you into conflating water permeability with active solute transport, because Nephron separates these mechanisms to generate medullary osmotic gradient. Question 1 reads, which segment of the nephron is impermeable to water but actively reabsorbs NaCl, thereby diluting the tubular fluid? The options are proximal convoluted tubule, descending limb of the loop of Henle, thick ascending limb of the loop of Henle collecting duct, and Bowman's capsule. The structural configuration of the loop of Henle relies on a stark functional asymmetry between its segment. Let's say this is Bowman capsule, then proximal convoluted tubule, then descending limb of loop of Henle, hairpin loop of Henle, then ascending thin limb and ascending thick limb of Henle. And finally it becomes DCT and it exits as collecting duct. While the thin descending limb permits passive water exit into the hypertonic medullary interstitium, the thick ascending limb features an epithelium that is completely tied to water molecules. This segment expresses the sodium-potassium 2-chloride co-transporter on its apical membrane, which utilizes the low intracellular sodium gradient generated by the basolateral sodium-potassium adenosine triphosphate pumps to drive ions out of the lumen. Because ions leave while water is trapped inside, the luminal fluid osmolarity drops significantly below the plasma level, which is precisely why this region is universally known as the diluting segment. So, the correct answer would be C. The proximal convoluted tubule is incorrect because it is highly permeable to water via aquaporin 1 channels, performing isoosmotic reabsorption where water follows solutes proportionally. The descending limb of the loop of Henle is highly permeable to water and lacks significant active sodium chloride transport, causing the tubular fluid to become progressively concentrated rather than diluted. The collecting duct is incorrect because its primary job is to either release water or absorb water based on hormonal orders, and its permeability is variable and tightly regulated by antidiuretic hormone via aquaporin 2 channels, rather than being constitutively impermeable and dedicated to diluting the fluid. Bowman's capsule serves as the initial filtration barrier, where the fluid remains entirely isoosmotic to plasma, making it completely incorrect. Moving forward, this brings us to how the body handles direct failure of these water channels down the line. Question 2 reads, a patient presents with polyuria. That means the patient is passing 3 to 4 liters of dilute urine per day despite adequate fluid intake. Plasma ADH levels are normal. And which of the following is the most likely defect? Options are insufficient ADH synthesis by the hypothalamus, failure of collecting duct cells to respond to ADH, excess aldosterone production, overactive NKCC2 co-transporter, and option E is increased GFR due to afferent arterial dilation. Okay, so to solve this question, let's talk about antidiuretic hormone. ADH is also known as vasopressin, is synthesized in the supraoptic and paraventricular nuclei of the hypothalamus and then stored in the posterior pituitary gland. Under normal physiological conditions, a rise in plasma osmolarity triggers its release, allowing it to bind to type 2 vasopressin receptors on the vasolateral membrane of the principal cells in the collecting duct. This binding activates a cyclic adenosine monophosphate second messenger pathway that inserts aquaporin 2 channels into the apical membrane to conserve water. When a patient exhibits severe polyuria with dilute urine despite having completely normal plasma hormone levels, the pathology is not central, it's peripheral and organ resistance where the principal cells fail to respond to the circulating hormone. That's why the correct answer is B. Option A says that insufficient antidiuretic hormone synthesis by hypothalamus, but this scenario describes central diabetes insipidus, which is completely contradicted by the fact that the patient has normal plasma hormone levels. Option C is wrong because excess aldosterone production would cause sodium retention and potassium wasting, typically leading to hypertension rather than the excretion of massive volumes of highly dilute urine. An overactive sodium-potassium 2 chloride transporter, mentioned in option D, would enhance medullary hypertonicity and increase water reabsorption capability, which contradicts the clinical presentation of profound polyuria. Increased glomerular filtration rate due to afferent arterial dilation could elevate urine volume slightly but would not selectively manifest as a specific defect in water concentrating ability while leaving hormone levels unaltered. Now let us pivot from water balance to the master hormone regulating systemic volume and pressure. Examiners love testing whether you can pinpoint the exact cellular coordinates where mineralocorticoids modify ion transport to alter systemic hemodynamic states. Question 3 reads, aldosterone increases blood pressure primarily by acting on which structure? The given options are glomerulus, proximal convoluted tubule, thick ascending limb, principal cells of the distal convoluted tubule and collecting duct, and option E is macula densa. To get the answer, let's understand the mechanism behind it. Aldosterone is a hormone synthesized and secreted by adrenal cortex. Let's say these are kidneys and adrenal glands are over the kidneys, kind of like in this structure. An adrenal gland has two parts, mainly one is cortex, the other one is adrenal medulla, and adrenal cortex will secrete aldosterone in response to elevated angiotensin 2 levels or high serum potassium because it is lipophilic. I'm talking about aldosterone. As aldosterone is lipophilic, it diffuses across cell membrane and binds to intracellular aldosterone receptors. And the receptor is located within the principal cells located in the late distal convoluted tubule and the collecting duct. Let's say this is Bowman capsule. We have proximal convoluted tubule, then we have loop of Henle, then we will have distal convoluted tubule, then collecting duct. The receptors will be specifically located here or here. Aldosterone, when connects with the receptor, it will cause transcription of specific proteins, leading to increased expression and activity of apical sodium channels called ENAC and basolateral sodium potassium pump. The net kinetic result is markedly accelerated sodium reabsorption from the lumen into the blood, creating an osmotic draw that pulls water along with it to expand extracellular fluid volume and elevate systemic blood pressure. So the correct answer would be D. Now let's check why other options are not correct. Option A, glomerulus, is entirely incorrect because it acts purely as a mechanical size and charge selective filtration barrier and lacks aldosterone sensitive transport machinery. The proximal convoluted tubule is responsible for bulk constitutive reabsorption of solute and water and is primarily regulated by angiotensin 2 and sympathetic tone rather than aldosterone. Option C, thick ascending limb, utilizes the sodium-potassium 2 chloride transporter to build the medullary gradient and is the target of loop diuretics, not the primary site of aldosterone-mediated pressure control. Option E, macula densa, acts as a specialized chemoreceptor zone within the thick ascending limb that senses luminal sodium chloride concentrations to regulate renin release via tubular glomerular feedback, making it functionally distinct. Understanding these localized pressures allows us to analyze the exact quantitative forces governing the initial filtration trap. When systemic oncotic pressures collapse due to nutritional deficits, the Starling forces across the fenestrated capillary walls shift instantly toward hyperfiltration. Question 4 regarding that reads, the net filtration pressure at glomerulus is approximately 16 millimeters of mercury. If plasma protein concentration drops significantly, for example due to malnutrition, what happens to the net filtration pressure? To unravel the answer, let's look at the underlying mechanism first. Glomerular filtration is dictated by Starling forces across the capillary wall, expressed mathematically as net filtration pressure equals glomerular capillary hydrostatic pressure minus Bowman's space hydrostatic pressure minus plasma oncotic pressure. The plasma oncotic pressure is the osmotic force exerted by non-filterable proteins, primarily albumin, which acts to hold fluid inside the vascular space and resist filtration. When severe malnutrition limits amino acid availability, hepatic synthesis of albumin drops precipitously. This substantial decline in plasma protein concentration directly lowers the plasma oncotic pressure, removing the major opposing force to filtration, which mathematically increases the net filtration pressure and drives excessive fluid across the barrier into Bowman's space. So the correct answer is B. For complete comprehension, we should also practice eliminating other options. Option A states that net filtration pressure decreases because plasma oncotic pressure falls, but it's a logical fallacy, as lowering an opposing force must always increase, not decrease, the net forward pressure. Option C, it says that it stays the same because autoregulation compensates. While renal autoregulation via myogenic mechanisms and tubulo-glomerular feedback works tirelessly to keep glomerular filtration stable during pressure shifts, it cannot fully neutralize a severe systemic drop in protein concentration. Option D, the hydrostatic pressure within Bowman's capsule does not arbitrarily increase in response to low systemic protein levels, as it is primarily governed by downstream tubular patency and fluid flow. Glomerular hydrostatic pressure itself is maintained by the relative tones of the afferent and efferent arterioles and does not increase as a direct chemical consequence of systemic protein loss. Now, to master renal pharmacology, you must realize that blocking solute transport in the medulla simultaneously disables the concentration gradient and floods the collecting duct with unreabsorbed water. Question 5 reads, a drug blocks the NKCC2 co-transporter in the thick ascending limb. What is the expected effect on urine output and medullary osmolarity? Options are decreased urine output, increased medullary osmolarity, increased urine output, decreased medullary osmolarity. Option C is decreased urine output and decreased medullary osmolarity. Option D says no change in urine output but increased medullary osmolarity. Finally, option E is increased urine output, increased medullary osmolarity. To understand this, we need to understand loop diuretics, including the generic molecule furosemide, serves as a prime clinical example of this mechanism. The loop diuretic class of medications, including the generic molecule furosemide, serves as a prime clinical example of this mechanism. The pharmacological action centers on the reversible inhibition of the sodium-potassium 2 chloride transporter in the thick ascending limb of loop of Henle. By preventing these ions from being actively transported out of the tubular lumen into the medullary interstitium, the drug directly obliterates the hypertonic medullary osmotic gradient, dropping it significantly below its normal peak level of 1200 milliosmoles per liter. Without this osmotic gradient, the collecting duct completely loses its driving force to reabsorb water via aquaporins, resulting in a dramatic, powerful increase in urine output. That's why the correct answer is B. Let's see why other options are not correct. Option A. Decreased urine output combined with increased medullary osmolarity describes the exact opposite loop diuretic, representing instead a state of profoundly diuretic hormone. A drop in urine output, decreased medullary osmolarity given in option C, is a contrary physiological profile. The washed out medulla inevitably forces water to stay in the loop, increases output. Option D suggests no change in output or any medullary osmolarity failed to recognize that solute transport in the thick driving the countercurrent multipliers. Increased urine output and increased medullary osmolarity, half correct and half wrong. Pay critical attention to the next question to protect your score. Examiners will love to phrase questions using the word except, trying to trick you into selecting a valid stimulus for renin release when they are actually asking you to identify the one option that suppresses it. Question reads, renin is released by juxtaglomerular cells in response to all of the following except, and there's 5 options: decreased renal perfusion pressure, sympathetic nervous stimulation, decreased sodium chloride delivery to the macula densa, elevated plasma sodium concentration. Option E is all of A, B, and C are correct stimuli. To answer this, you need to know juxtaglomerular cells are specialized granular smooth muscle cells located primarily in the afferent arteriole that act as the primary storage and release site for the enzyme renin. Renin release is tightly controlled by 3 classical synergistic homeostatic triggers. First is the drop in renal perfusion pressure, which acts as a direct intrarenal baroreceptor stimulus. Second, increased renal sympathetic nerve activity directly activates beta-1 adrenergic receptors on the juxtaglomerular cells. Third, a decrease in the sodium chloride delivery to the macula densa, signaling a low glomerular filtration rate, triggers chemical mediators to stimulate these cells. Conversely, an elevated plasma sodium concentration signals hypervolemia or hypernatremia, which acts as a powerful suppressive signal rather than an activator of the renin-angiotensin-aldosterone cascade. That's why the correct answer is option D. Now trying to eliminate other options. Option A, decreased renal perfusion pressure is a major valid physiological trigger for renin release, sensed directly by the afferent arteriole to protect the glomerular filtration rate. Option B, sympathetic nervous stimulation via beta-1 adrenergic receptors represents the neural pathway of renin activation during states of physiological stress, volume depletion, or orthostatic changes. Option C, decreased sodium chloride delivery to the macula densa cells trigger a chemical signal that lowers intracellular calcium in juxtaglomerular cells, directly promoting renin release. Because option A, B, and C are classic, well-established stimuli for the activation of this system, they're incorrect choices for a negative selection prompt. Now, from these systemic volume controls, let us zoom back into the highly specific transport limits of the proximal nephron. When analyzing biochemical transport thresholds, remember that finding a primary nutrient in the urine is not a failure of filtration, but an absolute saturation of the apical transport machinery. Question 7 reads, glucose is found in the urine of an individual. Which of the following is the most accurate explanation? Options are glucose is not normally filtered at the glomerulus. Option B, the collecting duct has failed to reabsorb filtered glucose. Option C is plasma glucose exceeds the transport maximum of SGLT2 in the PCT. Option D is ADH has increased glomerular permeability to glucose. And option E is the loop of Henle has secreted glucose into the tubular lumen. To get the answer, let us understand that under normal physiological conditions, glucose is a small, uncharged molecule that freely filters across the glomerular basement membrane. This is Bowman capsule, then this is glomerulus, and glucose freely filters across from glomerulus into Bowman's. That's why the concentration in plasma and Bowman's space is same for glucose. Under normal glycemic conditions, 100% of this glucose is completely reabsorbed in proximal convoluted tubule, primarily via sodium glucose co-transporter 2, located in the apical membrane. The co-transporter operates with a strict quantitative transport maximum, which averages approximately 375 milligrams per minute in healthy adults. When plasma glucose levels rise dramatically, the filtered load of glucose exceeds the kinetic transport maximum of these carriers, causing the transporters to become completely saturated so that the remaining glucose passes unabsorbed into the urine. That's why the correct answer is option C. The assertion that glucose is not normally filtered at the glomerulus is entirely inaccurate because its small molecular radius allows it to pass effortlessly through the filtration slits. The collecting duct possesses no specialized transport mechanisms for glucose reabsorption. Option B is incorrect, that's why. Antidiuretic hormone selectively modulates water permeability in the collecting duct via aquaporin channels and has absolutely no biochemical effect on glomerular permeability to organic solutes like glucose. Option E. The loop of Henle is highly specialized for countercurrent multiplication and ion transport, playing zero physiological role in the active secretion or handling of glucose molecules. When systemic metabolic acidosis threatens enzyme structures, the intercalated cells and proximal tubules must simultaneously dump protons and synthesize fresh bicarbonate to protect extracellular pH boundary. Having said that, let's read question 8. In metabolic acidosis, what is the renal compensatory response? The options are increased bicarbonate excretion and decreased proton secretion. Option B is increased proton secretion and increased bicarbonate reabsorption or generation. Option C is decreased ammonium production. Option D is inhibition of carbonic anhydrase in the PCT, and option E is decreased aldosterone release. Now, to solve this, let's ponder how kidney plays a definitive role in long-term acid-base compensation by adjusting the excretion of hydrogen ions and the reabsorption of bicarbonate ions. In a state of metabolic acidosis where primary systemic bicarbonate levels are low and pH is dropped, the renal tubules respond by accelerating proton excretion. In the proximal convoluted tubule, hydrogen ions are actively secreted into the lumen via sodium-hydrogen exchangers, while alpha intercalated cells in the collecting duct utilizes hydrogen ATP pumps to clear protons. Concurrently, the intracellular conversion of carbon dioxide and water generates new bicarbonate ions, which are transported across the vasolateral membrane into the blood to replenish the systemic buffer pool. That's why the correct answer is B. Increased proton secretion, both by proximal convoluted tubule and collecting duct, and increased bicarbonate reabsorption or generation. Option A says increasing bicarbonate excretion while decreasing proton secretion. This is incorrect because it would actively worsen metabolic acidosis by depleting the blood of its vital alkaline buffers and trapping dangerous protons in the body. Ammoniogenesis, option C, is actively upregulated during acidosis, allowing the proximal tubule to produce ammonium ions from glutamine, which serves as a crucial urinary buffer to safely trap and excrete protons. So saying decreased is incorrect. Option D, inhibition of carbonic anhydrase in the proximal convoluted tubule would completely block bicarbonate reabsorption, mimicking a type 2 proximal renal tubular acidosis and severely exacerbating the acidic state. Then, option E, a reduction in aldosterone release is incorrect because acidosis typically stimulates the adrenal cortex to release aldosterone, which supports proton clearance by upregulating the vacuolar hydrogen pumps in alpha intercalated cells. Now let us examine the fundamental physical constraints that dictate exactly what enters this entire system. The entire architectural purpose of the glomerular basement membrane is to establish a rigorous mechanical and electrical checkpoint that keeps structural proteins and cellular elements locked inside the systemic circulation. Question 9 reads, which of the following correctly describes the composition of glomerular filtrate compared to plasma? Options are given below, and to answer it, we need to talk about glomerular filtration barrier. The glomerular filtration barrier is composed of 3 highly specialized layers working in series. First, the fenestrated capillary endothelium, the negatively charged glomerular basement membrane, and the podocyte foot processes with their intervening filtration slits. This complex barrier acts as an ultrafiltration sieve that discriminates solutes based strictly on their molecular size and net electrical charge. Molecules with a molecular weight below 7,000 daltons, such as water, amino acids, glucose, urea, and electrolytes, pass through with complete freedom, ensuring their concentration in the initial filtrate are identical to plasma. Large macromolecules, particularly serum albumin and globulins, along with all cellular elements like erythrocytes and leukocytes, are completely excluded by size restriction and the strong electrostatic repulsion of the polyanionic basement membrane. That's why the correct answer is B, identical to plasma, except it contains no large proteins and no blood cells. Why the other options are incorrect? Saying that the filtrate is identical to plasma in all respects is completely wrong, because it ignores the absolute exclusion of vital cellular elements and high molecular weight serum proteins. The filtrate is not lower in glucose or amino acids initially, as these small molecules filter with an ultrafiltration coefficient of 1, achieving perfect osmotic equilibrium across Bowman space. The concentration of albumin in the filtrate is exceptionally low, approaching near zero values under healthy physiological conditions, because the charge and size barrier actively repels it. Describing the filtrate as containing only water and inorganic ions is a significant oversimplification that overlooks the massive pre-filtration of crucial organic solutes like glucose, amino acids, and nitrogenous waste products. Finally, let us explore how the heart communicates directly with these filtration barriers to manage volume overload. When the venous return stretches the right atrium, the cardiac muscle transforms into an endocrine organ to force the kidneys to dump volume and drop systemic vascular pressures. Question 10 reads, atrial natriuretic peptide is released in response to increased atrial stretch. Its net effect on the kidney is best described as, here are the 5 options. First one is increased sodium reabsorption and water retention. B is decreased GFR and aldosterone stimulation. C is increased GFR, decreased sodium reabsorption and diuresis. D, vasoconstriction of the afferent arteriole. E is stimulation of ADH release from the posterior pituitary. First of all, some facts about ANP, or atrial natriuretic peptide. This ANP is a polypeptide hormone synthesized, stored, and released by atrial myocytes. This is hard. We have atrium, one over here, another here. Atrial myocytes release this ANP, and this ANP is released in response to mechanical stretch, which signals an elevated intravascular volume state. Once in the circulation, this ANP targets the renal vasculature and also tubular segment to aggressively promote natriuresis and diuresis. It achieves this by selectively dilating afferent arteriole while constricting the efferent arteriole, a coordinated hemodynamic maneuver that significantly elevates glomerular capillary hydrostatic pressure and increases the glomerular filtration rate. Concurrently, it directly inhibits sodium transport in the medullary collecting duct. Let's say this is proximal convoluted tubule, then we have loop of Henle, then we have distal convoluted tubule and collecting duct. Collecting duct has 2 parts. One is cortical part, the upper one, and the other one is medullary part. And ANP inhibits sodium transport in the medullary collecting duct and suppresses the release of renin, aldosterone, ensuring that less sodium is reabsorbed and a massive volume of water is cleared into the urine to decompress the cardiovascular system. And that's why the correct answer is C. Increased GFR, decreased sodium reabsorption, and diuresis. Why are the other options wrong? Option A. Increased sodium reabsorption and water retention describes the exact physiological profile of the renin, angiotensin, aldosterone axis, which acts in direct opposition to the volume clearing effects of natriuretic peptide. Option B. Decreased glomerular filtration rate paired with aldosterone stimulation is incorrect because atrial natriuretic peptide works tirelessly to shut down aldosterone production and increase renal clearance. Option D. Vasoconstriction of the afferent arteriole would sharply drop glomerular capillary pressure and limit filtration, completely undermining the hormone's primary mandate to excrete fluid volume. Atrial natriuretic peptide actively suppresses rather than stimulates, and the release of antidiuretic hormone from the posterior pituitary to ensure that water reabsorption in the collecting duct is completely minimized. Thank you for your focused attention throughout this intensive renal system lecture. We covered the superficial, the deep, and the intermediate topics, everything in one lecture. You can consider this whole video lecture as your masterclass for renal system. And I can confidently say that I have touched up on every single topic that IMAT will or can ask on testing. But you need to practice questions, and at the same time, you need to be linguistically capable of understanding the questions ad hoc or instantly. And for that, you need lots of practice. And for that purpose, I have the question banks. I have the complete biology guide. If you want that, you can check that out. The link will be in the description. And if you have any types of question that you feel like you're stuck at, you can mail me and I will try to solve that for you. As you have reached at the end of this lecture, it's my promise to you that I will be helping you till the IMAT to pass the exam with flying colors. Your deep commitment to mastering these core medical principles from first principles is exactly what will set you apart on your path to top tier academic success. And to lock in these concepts and ensure you have all the anatomical and biochemical facts at your immediate disposal, practice as much as you can. Keep pushing your boundaries, stay disciplined, and I will see you in our very next high yield session. Those who fully watch the video, comment completed, and we will send you a special gift to you for your exam. And finally, drop a like, subscribe, comment if you have any question. Peace.